All right, the top of the hour is approaching, so I will just quickly get things started. Welcome everyone to Contributed Papers section number 1, Species Distributions in the Niche. I'll be the moderator. My name is Scott Egan. Our question wrangler will be Volker Rudolph, and our technical moderator and expert will be Mike Whitlock. As the moderator, I will be announcing each speaker and then letting them get started. I will signal to them using these wonderful pieces of construction paper that my daughter helped cut out at 16 minutes and then at 19 minutes. The 19 one will be a little bit more obvious. Moderators will then reactivate our own videos at 19:45 so that we can get the next speaker queued up. If you have questions, you may use the Slack channel for this specific section or the chat in the Zoom right now. And so without further ado, we'll get started with Erica Baken, who will be talking about arboreality correlates with climate across the family of lungless salamanders, Plethodontidae.
Oops. All right. Thank you, Scott. Like he said, my name's Erica Baken, and I'm going to talk about salamanders and the fact that arboreal and terrestrial salamanders occupy distinct climatic niches. This is part of my work that I did for my dissertation at Iowa State, and I'm excited. It's going to be fun. So to introduce y'all to the stars of the show, I've got a phylogeny here of all extant salamanders. Some familiar faces might be on the right. We've got sirens, tiger salamanders, hellbenders, and mud puppies. But what I'll be talking about today is the family Plethodontidae, the lungless salamanders, and that's this highlighted clade here. As you can see, it's about 60 to 70% of all extant salamanders, and as their name suggests, they're lungless, and so they breathe primarily through their skin. It has a couple cool quirks about it, but it has a lot of ecological consequences as well. Because they only breathe through their skin, it has to be very permeable, and so almost all these species are really susceptible to desiccation, and that'll come back into play a couple times throughout the talk. But what I found really interesting about this family in particular, especially at the beginning of my dissertation, is I started looking at microhabitat use across the family and was surprised to find that arboreality is really common and fairly widespread across the phylogeny. So all these green dots are species that spend most or all of their time living in trees. And it's quite surprising, mostly because of that desiccation characteristic that I talked about. And this really became kind of the backbone of my dissertation, figuring out why arboreality is so common and so spread across the phylogeny. What were the factors that pushed them up into trees and more frequently pushed them out of the trees again? So when considering what it takes to push species into or out of microhabitats, lots of things obviously could be at play. There could be some morphological constraints that open up different microhabitats to species or constrain them. Competition, predation, phylogenetic constraint, all of these things obviously could be driving microhabitat use. But for this project in particular, we honed in on the idea that climate could be determining the availability of different microhabitats for salamanders. And we did so for a couple reasons. First, we know that on much smaller scales, some ecological studies have shown that environment and weather play a pretty substantial role in driving short-term microhabitat use. So lots of evidence of species being driven around the landscape just by weather changes. That's fairly well determined in these salamanders. We also found in a different part of my dissertation that climate on this macroevolutionary scale can influence these salamanders. So I was able to demonstrate that across the evolution of these salamander morphology, the trait of surface area to volume ratio has been constrained by climate. So we find that species that live in climates that are generally drier, those species, their morphology has been constrained such that they have much lower surface area to volume ratios on average. So we know that climate can have this macroevolutionary impact on plethodontids. And we know that in the short term, weather is important for microhabitat use. So that led us to the hypothesis that climate impacts plethodontid microhabitat use on a macroevolutionary scale. So to get at this hypothesis, we took advantage of some great free online resources. We got species occurrence data from VertNet and did some verification procedures with other data sources like the IUCN Red List to find out where all these species are living. We extracted climate variables mainly from the WorldClim database, but some other weather-based data sources so that we have information on air temperature, precipitation, elevation, cloud cover, a couple other variables as well. And then we just classified as many plethodontid species into microhabitat types. And this was based on primary literature, field notes, and other sources like that. So with all these data, we took two separate approaches to get to our hypothesis. And the reason we did these two separate things is each of these approaches has some weaknesses and some strengths. And so we were hoping to get a little bit of a congruence between the two approaches. And so we did phylogenetic comparative methods, and then we also did some ecological niche modeling. For the phylogenetic comparative methods, we had climate variables extracted from all of those occurrence data that we had for each species, and we summarized them across the species. This was one of the weaknesses of the phylogenetic comparative method. We couldn't use the full climate data set. We had to do species summaries, so we lost some information there, but it's okay. And this resulted in data for 277 of our plethodontid species. And we just did a very simple phylogenetic ANOVA with a couple robustness analyses as well, just generally testing whether climate was a function of microhabitat use, while accounting for phylogeny, of course. For the ecological niche modeling, we were able to take advantage of all the climate data from across the species occurrence data sets, and which was an advantage that the phylogenetic comparative methods didn't have, but there were also some limitations with the modeling as well. For this data set, we trimmed it down to just arboreal and terrestrial species, so excluding all other microhabitat types. And then we also trimmed it down a little bit further so that we only were looking at species for which we had over 15 data points. And that is determined to be a good cutoff for the algorithm that we used. And this resulted in a data set of 183 species, so a little smaller, but still substantial. We used the max-ent algorithm, which is pretty common for our niche modeling, and we did species paired niche overlap measures. So for each species, we compared niche overlap with every other species in our data set. And I'll talk a little bit more about the full analysis when we get to it, but we were just using pretty common niche overlap metrics of Schoner's D and Warren's I. Again, I'll go back to a little bit more nitty-gritty stuff when we get to that. But first, I want to show you the phylogenetic comparative methods results. So again, just a simple phylogenetic ANOVA looking at climate as a function of microhabitat use. And this is the principal component plot of those data. All of these empty circles are species means. The filled circles are microhabitat means. And so we've got our arboreal species in green. This is the convex hull of all of our arboreal species in climate space. And then the black is the terrestrial species. And so we were looking at the comparison between arboreal and terrestrial. And I can talk about why we weren't so concerned with the other microhabitats later if you're curious. But this was our main test about arboreal versus terrestrial. And simple analyses showed that, yes, they in fact do have different climatic means when we look at all of the climate data together. And that was also corroborated when you just prune down the climate data to PC1 and PC2. We see significant differences between our arboreal and terrestrial species groupings. Now, to get more specific information on what exactly was differing between these two climates, we looked at the different climate variables separately between arboreal and terrestrial species. This is all those data together, but what I want you to look at in particular is this top left plot. These are 3 climate variables that correspond to temperature. For all of these, we see arboreal species living in warmer climates than our terrestrial species. Also of note is this top right bit where we have 3 different climate variables that represent precipitation. And we don't see any difference between arboreal and terrestrial species with respect to precipitation. Pretty surprising because the main driver of our hypothesis was related to desiccation risk. And if you live in a place with a lot of rain, you would assume that your desiccation risk is mitigated, but that didn't play out with these data. And the last thing I want to pull your attention to on this plot is this one down here. We've got elevation showing a significant difference between arboreal and terrestrial species, such that arboreal species live in lower elevations. Got another significant relationship over here, but that didn't bore out with our robustness analyses. So the ones that we found robustly coming out consistent across all of our kind of variations of our analyses was arboreal species living in warmer regions and lower elevations. And we've got some Bonferroni adjustments on the alpha values as well. So those are nice and robust. Overall, from the phylogenetic comparative approach, we've got arboreal and terrestrial species living in different climates that's characterized by arboreal species living in warmer regions in lower elevations. Onto the niche modeling bit. Before I give you the results from these guys, I'm going to quickly overview exactly what we were looking at, what we were comparing here. You can imagine on the left, we've got a distribution for some random species A. And what you do with the ecological niche modeling is you take the climate variables from where we know they occur, do some regression. And once you get a good model for what that climate generally is, you can project it across a larger geographic extent and get a sense as to where else we find similar climates. And so on the left, you can see all the places that are blue would be good places for this theoretical species A to live climatically. If climate was the only thing determining their distribution, but it's not, but you get it. So for the species comparisons, you take one projected model compared to a second projected model and simply just find how different those models are pixel by pixel. And what that does is it gives you a niche overlap score that ranges from 0 to 1, 0 meaning there's no niche overlap. These are completely dissimilar niches, and 1 meaning that they are identical niches. All right. So we've got our niche overlap scores for each species pair. A couple more steps. So one of the last steps that we did here was classify each of those species pairs as either a cross microhabitat comparison, where one species was arboreal, the other species is terrestrial. That would be a cross microhabitat comparison. Or if both species are terrestrial, that's a within microhabitat comparison. So for each species pair where we have a niche overlap score, it's either a cross microhabitat comparison or a within microhabitat comparison. All right. 2 more considerations for this model were phylogenetic relatedness. We wanted to include that information, of course. And then we wanted to take into account geographic proximity, because 2 ranges, even if they're not overlapping, if they're close to each other, they're gonna give you a higher degree of niche overlap than you would expect by chance. So we wanted to account for geographic proximity in our model as well. And so we just have those values for each of those species pairs. And again, our statistical model was fairly straightforward. Is niche overlap determined by microhabitat comparison type while accounting for phylogenetic and geographic distances? So what we expected out of these results, if climate does drive microhabitat use macroevolutionarily, we would expect that the cross microhabitat comparisons would show the least amount of niche overlap. One step further, if we think like we did, that arboreality in particular is constrained by climate, we would expect that the arboreal comparisons would have overall a greater amount of niche overlap. So again, our predictions were that the cross microhabitat comparisons would have the lowest amount of niche overlap, and the arboreal comparisons would have the most amount of niche overlap. And that's what we found. This is the histogram of one of the metrics that we used, shown as D, but it's consistent with the other metric we found as well. Here we have in brown the cross microhabitat comparisons of arboreal versus terrestrial. And as you can see, the mean of these values is lower than the other two means. In black, we have our terrestrial comparisons, and in green, we have our arboreal comparisons. And looking at the pairwise statistics, we find that each of those groupings are significantly different from one another, with fairly large effect sizes associated with each of those pairwise comparisons, aligning nicely with what we predicted. So bringing both of our analyses back together to get some broader conclusions, we found from both the ecological niche modeling and the phylogenetic comparative methods that arboreal and terrestrial species both live in different climates. The phylogenetic comparative approach allowed us to show that those differences are largely driven by warmer temperatures and lower elevations for our arboreal species. And lastly, with the ecological niche modeling, we were able to show that there's more niche overlap between arboreal species than there is across those microhabitats. So arboreal species display more niche overlap with other arboreal species than with terrestrial species. So all these things are cool and exciting, and they all align with our hypothesis that climate impacts plethodontid microhabitat use on a macroevolutionary scale. I will caveat this whole thing to say that there are several other pieces of evidence that would definitely help lend more confidence to this conclusion. It's hard to really close the book on this problem because the pinpointing of exactly when the microhabitat transitions happened phylogenetically and geographically is a little fuzzy. That would be great information that we could use to test this hypothesis even further. Other things as well. All that's to say, all of our evidence is consistent with our hypothesis. However, there could be other studies that help lend more confidence or disprove this in the end. But we can talk about that if you're interested. Now with that, I would like to thank my co-authors, Lauren Mellenthin, and my PhD advisor, Dean Adams, and then my Iowa State lab mates, Elizabeth and Brian. Everyone was really great helping with this project. And I think I've got a handful of minutes for questions, and I would be happy to answer them.
All right, we have one question coming in here from William Godsoe asking, how frequently do plethodontists revert from arboreality to terrestriality? Kind of like, how do you see that switch?
Yeah, so macroevolutionarily, a lot. So one of the other chapters in my dissertation showed that we've got somewhere between 5 and 7 transitions towards arboreality and somewhere around 60 reversions back to terrestriality. So it's a really uneven distribution. It seems like the abandonment of arboreality is way easier. And so that is another reason why we were pinpointing climate as a potential driver. If it can drive you out of the trees, that would align nicely with how frequently we see that microhabitat being abandoned. There are other species, though, that are kind of semi-arboreal, that spend part of their life in trees and part of their life on the ground or underground. And so there are shades of how you would classify arboreality. And so some of our robustness analyses that we did was toying around with how exactly we cut off the definition of what is an arboreal species versus not.
Well, while we're waiting, Stephen, to ask one, I'm going to abuse my moderator power by asking a question myself. So, I mean, it was very much reminded for like another amphibian group's frogs, where you also have the terrestrial and so like the tree frogs. Like, do you see similar patterns in both of those groups? Do you know, or are they different in some aspects?
I am not sure if we've gotten anyone to study climate and arboreality in this way. I know that frogs are far more successful as arboreal species. There are a lot of arboreal salamanders, but frogs really were able to turn that into a very useful niche, where it seems like with the salamanders, there wasn't quite the adaptation to make arboreality a really fruitful niche for them in terms of macroevolution. We don't see toe pads evolving, so we don't have like specialized really good climbers or anything for salamanders. Even the climbing performance from species to species doesn't seem to be better in arboreal species. So it's still a large question as to what they're doing up there and why they went up there in the first place, but it's a little certainly different than frogs.
We should probably get moving on to our next speaker. Next up, we have a pre-recorded talk from Yingtong Wu, titled Climate Niche Position and Post-Glacial Accessibility Determine Oak Species Range Size in North and Central America.
This is the first video of the meeting, so please let me know if the sound doesn't come through or something.
Hi, everyone. It's my pleasure to be able to talk here in this virtual event. My name is Amanda Wu from the biology department of University of Missouri in St. Louis. Today, I'm going to present one of my chapters in oak species range size. And I want to explore how species range size is influenced by niche property and post-glacial migrational legs in the Americas. So first, I'm curious on the question, why are some species widespread but some are rare? This question can be dated back in the origin of species by Darwin, when he asked why one can explain who can explain why one species ranges widely and is very numerous, and why another allied species has a narrow range and is rare? And this question is still important nowadays because knowing why some plant species are rare is important for conservation. And there are basically two pathways to explain species range size. First is the amount of suitable habitat that a species can occupy. This could be explained by a lot of different mechanisms, as shown in this arrow here. Most of the explanations are related to niche breadth and niche position, which is an intrinsic properties of a species. And the second pathways is showing down here, occupancy of suitable habitat, or in other words, range filling. Not all of the species can occupy 100% of the amount of suitable habitat, and this percentage of occupancy can also contribute to the species range size. So I'm going to focus on 4 hypotheses that are going to contribute to the variation of species range size. The first is the niche breadth hypothesis. And I have this cartoon showing on the left here to explain the mechanism. So for the rare species, they will have narrower niche breadth. So the blue grid here shows that it can occupy only a narrow subset of environmental variables. That's why they can only occupy a small number of grids in the current landscape. But for the widespread species shown down here, it can occupy a more larger range of environmental variables, and that's why it can occupy a higher amount of suitable habitat, which contribute to more species range size. So the prediction of this hypothesis is a positive relationship between species niche breadth and the amount of suitable habitat. And this has also been one of the most tested hypotheses in a lot of literature. And for the second hypothesis, we have the niche position hypothesis. So this also focuses on species niches and the amount of suitable habitat, but it's looking at from a different perspective. So here, again, from the cartoon here, rare species occupy a more marginal or uncommon type of suitable habitat. But for the widespread species, it occupies a more common, a more typical environmental habitat. So even though for the rare and widespread species, they have the same amount of niche breadth, they have very different suitable habitat because of this niche position. So here we'll predict a negative relationship between species niche marginality, meaning how marginal your niche position is, with the amount of suitable habitat. Compared to the niche breadth hypothesis, niche position hypothesis has much less tests, empirical tests, but it's still an important hypothesis that needs to be tested. For the third hypothesis, we're looking at species range size from a more different perspective. We're looking at the colonization ability of a species. So this is very straightforward, meaning that more widespread species we will expect a high colonization ability. For more localized or more narrow range species, we will expect a lower colonization ability. So colonization ability can be represented by a lot of different intrinsic properties of a species, including dispersal distance, the number of propagales, survival rate, etc. And we'll predict a positive relationship between colonization ability and the occupancy of suitable habitat. So pay attention to here, it's going to contribute to the second pathway instead of the first pathway. And this is a popular hypothesis in the species range size literature. And then for the fourth hypothesis, we're going to focus on a more dynamic or historical perspective. For a lot of present species distributions, they are not only influenced by the current climate or current niche properties, but also the climate in the last glacial maximum. So here I'm looking at the connectivity or accessibility of suitable habitat from last glacial maximum to the current landscape. For rare species, we might expect a higher disconnection between last glacial maximum suitable habitat and the current suitable habitat, and that can contribute to a more narrow occupancy of suitable habitat. And for the widespread species, we might expect a higher connectivity between last glacial maximum habitat and the current suitable habitat. So in other words, we predict a positive relationship between the post-glacial accessibility and the occupancy of suitable habitat. This hypothesis has been supported by a lot of European studies, but there's not a lot of empirical tests for American flora. And for the second hypothesis, for the second prediction of the same hypothesis, we will expect that the percentage of glacial ice sheet in the currently suitable habitat, as represented by these gray grids here, can influence the realized species range in the current landscape, because the occupancy of last glacial maximum ice sheet might influence the soil properties, mycorrhizal properties in the current landscape, and that could potentially inhibit the expansion, the range expansion of the species. So we expect a negative relationship between the percentage of last glacial maximum ice sheet in the current landscape and the occupancy of suitable habitat. So I'm done talking about this 4 hypotheses, and I'm going to show you the study system I'm going to use. I'm going to focus on the oaks in Americas for several reasons. So first, it's because they have a very high diversity of oak species in North and Central America here. This gives me a lot of statistical power to test all these 4 different hypotheses. As shown on the left figure here, the phylogeny of all these species have also been figured out quite clearly, so we can take into account the phylogenetic effects. For the second reason, there's a great variation in species range size and species niches among all these different species. So the great variation of this niche properties can help me to explain this different hypotheses. So for the methods, I'm showing this diagram to explain the databases I'm using and the methods I'm using. I'll have 183 oak species occurrence points from the BN data sets. And I also got 19 climate variables and 10 edaphic variables downloaded from the different sources. For the 183 oak species occurrence points and with all this climate and soil variables, I'm able to use the Maxent species distribution modeling to calculate or quantify the current suitable area. And I also use Maxent algorithm to calculate what was the last glacial maximum suitable area for each of the species. So from the current suitable area, I'm able to use outlying mean index analysis to calculate the niche breadth and the niche position for the first and second hypothesis respectively. And from last glacial maximum suitable area and current suitable area, I'm able to calculate the post-glacial accessibility and the last glacial maximum ice sheet percentage in the current landscape. So this is for the 4th hypothesis regarding the post-glacial migration lag hypothesis. And for the 3rd hypothesis on the colonization ability, I used Kevin Bear's data from the paper, and I also used some of the trait database as well as the FIA tree database to extract some functional traits, tree maximum heights, and the dry seed mass to represent the colonization ability of oak species. And last but not least, with all these occurrence points for each species, I'm able to calculate the species range sizes. I used 2 different quantification for range sizes. One is the area of occupancy, the other is extent of occurrence. For this talk, I'm only gonna focus on the area of occupancy because of the limited time. Just more about the methods. I want to explain a little bit how I calculate the post-glacial accessibility. It's basically about the distance between the current suitable habitat and the last glacial maximum suitable habitat grades. So on the left figure here, we might have a very high disconnection between these 2 subsets of suitable grades. This high distance would transfer to the low post-glacial accessibility because we calculated the sum of inverse distance between these 2 sets of habitats. When you have overlapping or low distance between these 2 subsets of grids, the post-glacial accessibility would be high. So I'm done with the methods. I'm gonna explain what my results are. Just to recap, these are the expectations for explaining the amount of suitable habitat for each species. We expect that the climate niche breadth and soil niche breadth contribute positively to the amount of suitable habitat, and climate niche marginality and soil niche marginality contribute negatively to the amount of suitable habitat. And in return, amount of suitable habitat should be positively related to the species range size. As shown with this figure here, I use structural equation model for this relationships for all this 183 species. This diagram here shows the orange numbers are the standardized coefficients, and the asteroids here show the statistical significance for each of the pathway. As we see here, consistent with our hypothesis or the predictions, climate niche breadth is positively related to area of suitable habitat, but soil niche breadth is not related to area of suitable habitat. Both climate niche marginality and soil niche marginality down here are negatively correlated with area of suitable habitat. And all these three variables explained about 70% of the variation here. And at last, we see that area of suitable habitat is positively related to the area of occupancy or species range size. However, when we compare this observed coefficients with the expectation from spatial null models, we see that that doesn't deviate from spatial null models. Just in a larger perspective, we also want to see how the range size is explained by the occupancy or range filling in of the suitable area. And we actually see that there's only 52% that's explained by the occupancy of suitable area, and 55% of variation explained by the area, the amount of suitable habitat. So they kind of equally split among these two different pathways. For the range filling, we want to test how post-glacial migration lag and colonization ability affect range filling. First, by looking at this variation partitioning map, we see that a lot of variation are actually explained by post-glacial accessibility and last glacial maximum ice sheet percentage, but only 3% is explained by the leaf trait. So post-glacial accessibility has a much higher variation explained for occupancy of suitable area. And as we can see here on the diagram, this is consistent, the result here is consistent with our expectation. So higher post-glacial accessibility correlated with higher occupancy of area, lower percentage of last glacial maximum ice sheet correlate with higher occupancy of suitable area, etc. And when we look at the leaf trait parameter per leaf area, after variable selection in the model, we see there's no significant relationship with the occupancy of suitable area. And we also, after phylogenetic, adjusted for phylogenetic relationship, there's still no significant relationship. So just to summarize the results, we see that for the first pathway, explaining the amount of suitable habitat, we see climate niche breadth contributes positively to amount of suitable habitat. Climate niche marginality and soil niche marginality, both of the niche positions properties, they contribute negatively to the amount of suitable habitat. And in return, the amount of suitable habitat is positively related to species range size. And for the second pathway explaining the occupancy of suitable habitat or range filling, we see that post-glacial accessibility and percentage of last glacial maximum ice sheet contribute to the variation of occupancy of suitable habitat, but in different directions. So higher post-glacial accessibility contribute to higher occupancy of suitable habitat, but higher last glacial maximum ice sheet percentage contribute negatively to the occupancy of suitable habitat, and others are consistent with our hypothesis. However, colonization ability represented by traits are not positively related to occupancy of suitable habitat. So to summarize, the take-home message here is that widespread species occupy a more central or common niche in a larger range of suitable habitats. And second, widespread species have higher accessibility to current climatically suitable habitat from the last glacial maximum refugia, while rare species have lower accessibility. If you have any questions, please feel free to email me or put your question in Slack. I apologize, I cannot be present in this event. So if you have any questions, feel free to leave me the questions in email or in Slack. Thank you.
All right, thank you, Yingtong. It looks like we filled up the 20-minute slot there, so we're gonna move quickly on to Sebastian Schreiber, who will be our next speaker on coevolution of habitat selection in stochastic environments.
Great, thank you. Good afternoon, everyone. I'm going to begin with a quote from someone who introduced a key idea in habitat selection theory. And they wrote, "I was studying earthworm brains for my doctoral dissertation. I was irritated by Lack's dogmatic position that territorial behavior did not affect habitat selection. In desperation, I put it all into mathematical models and made several wondrous discoveries. I soon dropped the earthworm research. Both the worms and I were having nervous breakdowns and getting nowhere. Indeed, these were the words of Stephen Fretwell when reflecting on his paper with Lucas about the ideal free distribution, which states that for species living in a spatially heterogeneous environment with different patches, the per capita growth rate will be equal in all occupied patches and lower elsewhere. Now, intuitively, this sounds like an uninvadable strategy, but remarkably, it was not shown to be an evolutionarily stable strategy until the 21st century. Now, there's several important implications from the ideal free distribution. First, under equilibrium conditions, there should be no sink populations. Second, if the source of spatial heterogeneity is availability of resource input, there should be input matching. Then it also has implications for interacting species spatial distributions. If you have two competing species, there should be selection for complete spatial segregation of each species into the habitat where it's competitively superior. For predator-prey interactions, it can lead to conditions where you expect selection for contrary choices of the predator and the prey, which in the extreme case, that corresponds to enemy-free space. However, we know in nature, we see sink populations, we see resource input mismatch, we see co-occurring competitors. So we could ask, why is this happening? Of course, it might be that things are not being selected for it, or it might be, according to theory, that temporal environmental variation plays an important role, just as spatial variation does. And that can select for sink populations and input mismatch, and in the case of competing species, help exercise the ghost of competition past. So the goal of the work that I'm going to talk about is to sort of create a framework to understand the following question. What effect does spatial temporal variation have on the coevolution of habitat selection in many meta-communities? So I'm a theoretician, so I like to work with mathematical models. And the models I like to work with adhere to the KISS principle. Keep it simple, stupid, because when it comes to the complexity of nature, I'm inherently stupid. So in what ways am I going to adhere to the KISS principle? Well, in the models I'll be describing in the next slide, space will be implicit. The dynamics between the species are governed by mass action types of interactions, so Locke-Volterra dynamics. And the environmental fluctuations will be given by log Brownian noises. Okay, so let me tell you a little bit about the model. The details are not critical, but it's going to be a system of stochastic differential equations that can represent any number of patches in the environment, any number of species, and for each of the species, you're keeping track of their regional density. And then the patch allocation strategy, i.e. what fraction of the species is in each patch, is the quantity that's under selection. And the environmental fluctuations in time are given by, on the log scale, Brownian motion with any type of covariant structure. So what I want to do for these models is identify coevolutionary stable strategies, i.e. if each species is playing some type of patch allocation strategy, it cannot be invaded by mutants. So how do I define, in this context, these coevolutionary stable strategies? There's two ingredients. The first thing is think about some resident community of species where each species has some patch allocation strategy. We would like to know, do those species even coexist with that strategy and those types of interactions? So to check for whether or not the species coexist in the model, I use a criterion that was originally developed by Joseph Hoffbauer for deterministic models that's been extended to these types of stochastic models. And the beauty of this Hoffbauer condition is that it reduces the problem of checking coexistence to solving systems of linear equations and a linear programming problem. So that means it's analytically and computationally very tractable. Okay, so now we have a resident community where each species is playing a patch allocation strategy. What do we mean by those strategies being coevolutionarily stable? Well, it should resist invasion attempts within any species where you have a subpopulation playing a different patch allocation strategy. And you can write down the dynamics for that mutant subpopulation and then identify the stochastic growth rate when rare of that mutant population. That's actually given also just by solutions of linear equations. And if that quantity is negative, it means that invasion attempt by that mutant subpopulation of a given species will fail. So in this context, if you have this resident community where each species is playing a patch allocation strategy, that collection of patch allocation strategies is coevolutionarily stable if the invasion growth rate of any mutant strategy of any species is negative. Okay, that sounds all well and good, but how do you identify coevolutionary stable strategies for any given spatial / meta-community context? Well, it turns out, quite remarkably, there's a characterization of the coevolutionary stable strategy that relies on 4 key quantities. So I'm going to describe those quantities in the next slide. So let's focus on one of the species I in this meta-community. That species in each patch is going to experience fluctuations in its per capita growth rate. And those per capita growth rates locally, when you take into account the patch allocation strategy, determine the meta-population per capita growth rate of the species. Now, associated with these fluctuating per capita growth rates, there's 4 quantities of interest. So let me focus on one of the patches and at the same time keep track of the meta-population per capita growth rate. So the first pair of quantities I'm interested in will be the average across time, the mean per capita growth rate within a patch. So that's lowercase m. And then I'll also want to know at the meta-population level, what is the mean per capita growth rate across time. But the means are not enough. It's a fluctuating environment. So we'll also care about the variance of these fluctuating per capita growth rates. So what one needs to determine the third quantity is for each patch, you need to understand its covariance in its per capita growth rate with the metapopulation per capita growth rate. It turns out the weighted combination of those covariances determines the variance at the metapopulation scale in the growth rates for species I. So now I'm ready to characterize the co-ESS in terms of these four basic quantities. The first aspect is just the species that we're looking at is at stationarity. And so that means the stochastic growth rate of that species at the metapopulation scale, which is given by its mean minus 1/2 the variance, has to be equal 0 because you're at stationarity. The next thing is that all the occupied patches by species I, there's a balancing of two things. There's a balance of the contribution of that patch to the mean metapopulation growth and the relative contribution of that patch to the variance of the metapopulation growth rate. Those two relative contributions have to be perfectly balanced, i.e. the good, having a higher mean growth rate, has to be balanced with the bad, having a higher contribution to the variance. In unoccupied patches, the contribution to the variance has to be greater than the contribution to the mean. And those are patches that will not be occupied at the co-ESS. Now, there's several implications of these inequalities. The first one is what I said already, which is in occupied patches, the relative contributions to the mean and variance of the metapopulation growth rate are perfectly balanced. Second, if we were curious, when might we get the classical ideal free distribution where the mean per capita growth rates are equal in all occupied patches? That can happen in a variety of ways, not just one way. One way is that there's perfect buffering. In other words, the patch allocation strategy makes all the variance across the patches cancel out at the regional scale. Alternatively, if you have perfectly correlated fluctuations across space, at least in the occupied part of space, that can also lead to the classical ideal free distribution. Another implication of these inequality is if you have a highly buffered patch, it's going to be occupied unless its mean per capita growth rate is sufficiently negative. And finally, by the definition of the co-ESS, all the patches will have negative local stochastic growth rates, i.e. be stochastic sinks. Okay, so that's the characterization of the co-ESS. And if you think about this stuff a little bit, you might say, oh, this sounds like something relevant to economics. In other words, if I replace populations with wealth, there should be some microeconomic theory that's also related to this. It turns out the answer is yes. So the modern portfolio theory, which was discovered by Harry Markowitz, who won a Nobel Prize for his 1950s work on this, is related to what I just described. In other words, the co-ESS can be described as a tangency portfolio that lies on the efficient frontier, where the tangency is with respect to a risk-free asset with 0% return. And I'm sure that made things much clearer. Okay, what are some implications of this co-ESS criterion or inequalities to specific scenarios? So I'm going to look at two scenarios that correspond to antagonistic interactions. The first one is where both species are harmed by the interactions. So we're looking at competition. And I look at a scenario that's highly simplified, keeping with the KISS principle. So this will be where there's an even number of patches. In half of the patches, the orange warbler is competitively superior. In the other half of the patches, the yellow warbler is competitively superior. And that competitive superiority is being determined by this difference in the intrinsic rates of growth, which I'm denoting delta R. Now, and then the other thing I'm assuming, consistent with the symmetry, is there's the same amount of variation in time in each of the patches, and that the patches are spatially uncorrelated. Under these assumptions, you can easily write down what holds for the co-ESS for patch selection. And you can also understand when do you have different types of spatial distributions for these competing species. So the first thing that you can show is that if the variance at the local scale is sufficiently low relative to the difference in the intrinsic growth rates, then you get the ghost of competition past. There's selection for complete spatial segregation where each species only uses the patches in which they're competitively superior. However, if the local variance in temporal fluctuations is high enough, you get a partial exercising of this ghost of competition past, where species will co-occur, but and they'll increasingly look more even across the landscape as the amount of environmental variation increases in the landscape. What about another type of antagonistic interaction between predators and their prey? So in this example, which is the last example, I'll have a spatially asymmetric scenario where there's a source patch, which has environmental fluctuations, and a sink patch, which is very stable with little environmental fluctuations. In this case, again, you can use the criteria for the co-ESS to find analytic solutions for the coevolutionary stable strategy for the prey and the predator. And what it shows is, if we look at this left graph, if the environmental fluctuations in the source patch are sufficiently low, both species are selected to just use the source patch. If you have slightly higher variation in the source patch, it selects for just the prey to make use of the sink habitat, so you get an enemy-free sink. At higher levels of environmental variation, there's selection for the species to co-occur in both patches. In particular, you get a predator-prey sink population. And then finally, at higher levels of environmental variation, but not so high that species go extinct, you actually get selection for the predator to only use the sink habitat, and therefore you get an enemy-free source habitat. Okay, so now let me wrap up. So what I've shown you is a keep it simple, stupid framework that extends prior work on evolution of habitat selection to meta-communities experiencing spatial and temporal variation. A key idea in this theory is that the coevolutionary stable strategy in occupied patches balances local contributions to the mean of the metapopulation per capita growth rate and the variance of the metapopulation per capita growth rate. And then some of the consequences of this condition is that temporal variation can synergistically interact with species interactions to select for sink populations in a variety of ways. Of course, I've only touched on the tip of an iceberg of possibilities here. In particular, I've used very special spatial configurations for the examples and only considered two species interactions for the special examples. And it'd be interesting to know what happens in more realistic spatial configurations in more diverse communities. And of course, ultimately, it'll be even more interesting to understand what happens if you explicitly model space and restrict the way that the organisms can move across the landscape. So with that, I want to thank you for listening, ASN for hosting this delightful virtual Asilomar conference, and the National Science Foundation for funding some of this work. And I'll happily take any questions there's time for.
All right, I think we have some time for question, and William has a question, and he's asking saying that this analysis of stochastic growth rate looks really similar to Cheston's 2000 analysis of coexistence using an approximation of invasion growth rate. Do you know whether those are linked? And Cheston emphasizes a Taylor-Series approximation instead of covariances.
So it's linked in some sense. I mean, when you do the Cheston type of analysis of invasion growth rates with small noise, you're using what I would call a diffusion scaling. So you're making sure that the mean and the variance scale the right way when the noise gets low. And anytime you start doing that type of diffusion scaling, you often can write down a stochastic differential equation that approximates the original system, the original model. So in that sense, those diffusion type scalings are related to stochastic differential equations, which of course are related to this work. One way it is different is that these are not approximations of the growth rates. These are exact expressions for these models. So there's no approximation, it's precise. And importantly, it's analytically tractable because you're just solving linear systems and in some cases solving a linear programming problem, all of which things can be done by hand with perhaps a lot of effort or certainly done easily with computers.
Either everybody's busy typing or still thinking.
What was that about? Tangency portfolio, what in the world? Well, I can see the time's running out, so thank you again for listening, and feel free to contact me by email if you have any questions or comments. I'm still writing up this manuscript and we appreciate any feedback I can get.
And I was going to add, there's like, if you have any questions, like pop up afterwards, I always just like send them to me in the chat and I can post them in the chat. And so I give the speakers a chance to respond afterwards. So I keep an eye on the chat.
Great, thank you very much, Sebastian. Let's see, at the top of the hour, in about 30 seconds, we'll have Xiaoli Dong, who will be presenting on coupled evolution of landscape geomorphology and species traits across environmental gradients. Take it away, Xiaoli.
Right. Okay, thank you. Thank you everyone for zooming. What I'm going to talk about is the coupling between plant niche construction and the development of biogeomorphic landscapes. Landscape of different types of interesting morphology can certainly arise in places where there's no life. And this picture that you are seeing is a landscape on Mars. And there's no life, as we know so far, and then there's interesting morphology. But on Earth, we have all kinds of life forms and they can influence the landscape patterns. And this is an area that is recently developed in geomorphology called biogeomorphology. It studies the effect of plants on the landscape development. Here are some classic examples of biogeomorphic landscapes. The top left is the sand dunes and plant. These beach plants can influence the size and shape of these sand dunes. In fact, different types of the plants can shape different forms of sand dunes. And the other three pictures show some fluvial landscapes influenced by plant. Almost all of the biogeomorphic models that I'm aware of they assume that the plant that is doing these niche construction activities on these landscapes, they stay the same over thousands or sometimes tens of thousands of years. And they're doing same kind of activities, same type of species, which is not true as we've learned from niche construction theory. So niche construction theory studies the effect of the niche construction activities on the evolutionary dynamics of those plants that are carrying out those activities. So the essence of niche construction theory is this feedback loop. So you're starting with niche construction activities, then they will change the environment, the physical environment, and then that's going to change the selective environment of these plants, which will then influence the dynamics of the gene frequency for this plant population, which will then change the evolutionary dynamics for these geomorphic traits that are carrying out those niche construction activities. So this is a feedback loop that's studied in niche construction theory. However, this whole framework of niche construction theory has never been incorporated in the framework of biogeomorphology. So my background is from geomorphology, and what I try to do in this study is to put these two frameworks together. And the overlapping component of these two frameworks is this thing called plant niche construction activities. Geomorphology is studying this thing influencing the landscape patterns, and niche construction theory is looking at the effect of this activity on the evolutionary dynamics. So what I'm interested in is, what if we let the species, the plant species on this landscape, to evolve over time? And how would that influence the geomorphology of the landscape, as well as the development of those landscape over time? And another question is, that by putting these two frameworks together, it might also bring some new insights to niche construction theory, because a lot of niche construction theory assumes that the environmental change is only brought about by plant niche construction activities. There is no external environmental change. Some of the niche construction models does include independent environmental change, but it is often assumed very simplistic, like linear or periodic environmental change. However, a lot of especially in landscape evolution, a lot of physical processes are a lot more complex than linear changes or periodic change. So that's one aspect. I want to incorporate more realistic physical process into the niche construction theory framework. And second, for biogeomorphology, a lot of biogeomorphic landscape are self-organized. What that means is that local interactions can give rise to spatial patterns at global scale. And this global scale is part of the environmental change. This global scale pattern is part of the environmental change for these plants. So what about if we also include this feedback into the niche construction theory? How would that influence our understanding of the evolutionary dynamics of these niche construction traits? So this is the motivation of this study. And the question is, how do geoevolutionary feedback influence two things? First, how that's going to influence evolution of traits. And second is how that's going to influence the landscape development. I'm looking at two types of traits. First is the geomorphic trait, which determines the plant's capacity to change landscape geomorphology. And then second trait is, I call it ecological trait or recipient trait, which determines the plant's preferred environmental conditions. And so I'm following Sebastian's KISS principle, keep it simple and stupid. And my model is also very simple. It has only 4 state variables, the 2 ecological, 2 traits, 2 alleles corresponding to the 2 traits, ecological trait or recipient trait, and the geomorphic trait. And the third variable is the elevation of the landscape. And then the last variable is biomass. The model only has two parts. The first part, I have a module to describe the landscape evolution. So this is the governing equation of the landscape evolution. I have the baseline geomorphic rate change. And then second term is the effect on the elevation change from the plant niche construction. Then this term here is describing a saturation effect, meaning that when the change of the elevation will reduce as elevation increases. So at some point, it's going to reach some equilibrium state. And then this term here is called a long distance negative feedback. The kind of landscape that I have in mind is fluvial landscapes. And in a lot of fluvial landscape, imagine like a slow flowing river, and somewhere in that river, there's some plant patch growing. When the plant develop in that area, it's going to reduce the flow rate, it's going to reduce the shear stress and erosion. But in the area that's nearby that plant patch, the shear stress and erosion is actually going to be intensified because the existence of that plant patch. So that causes a long distance negative effect. So I have a set of equation goes into this S to capture that effect. And the last term is just a diffusion term, which to describe the sediment diffusion on the landscape. So this is a governing equation to describe how landscape is evolving over thousands of years time scale. Now, the second part is the evolutionary part. So I'm looking at plant populations. Each plant has several features. First, each plant has its own biomass, its own age, its own genotypes. And each genotype, I'm looking at 2 traits. So 2 alleles for 2 traits. I have the genotype for the recipient trait, and I have the genotype for the geomorphic trait. This is the equation to describe the growth of the plant, the change of the biomass, and the key thing here is that these two terms. The first term describes the environmental selection on the plant growth. The key thing here is this term, the difference between the allele value of this recipient trait and which defines the preferred elevation that plants like, and then this H is the actual elevation. So if these two values match, it means that plants are in a perfect place that they can grow happily, and then the biomass is going to be, the biomass change rate is going to be really high. But if the difference between these two values is really large, then the plant growth rate is going to be small. And the second term here is just to account for the cost of the niche construction. So this is the biomass. Then plant has its own life cycle. It's going to die at some point, and then they will produce some seeds before they die. And which seeds get to land on a vacant site is determined by three components. The first component is just the distance between two sites. Second component is how many seeds this plant can produce. If you produce more seeds, your offspring is more likely to take that site, right? So that's simple. Last term here is just how fit the match between the seed and the landing, the environmental condition of the landing site. And again, you see this, this difference between the R and H. So if the seeds prefer elevation of 1.3 meters and the landing site happens to have elevation of 1.3 meters, then the rate, the probability for that seed to successfully germinate on that site is going to be really high. So this is a dispersal process. So that's basically the whole model, which has these two parts. Now let's get to the result. What I'm going to show you a result in two parts. First is on the evolution of these traits, and second is on the how this geo-evolutionary feedback are going to change the landscape morphology. So let's first look at the evolution of the traits. What you are looking at here is the first trait, which is the recipient trait. So the recipient trait defines what kind of environmental condition the plants prefer to grow and so here it is looking at the elevation. What you are looking at here on the plot on the left is two lines. The red line is the landscape mean value of the recipient trait, and the blue line is the mean elevation of the landscape. At the beginning, the landscape is really, the elevation is really low, and the recipient trait evolves to a lower value to try to match with the evolution, the external condition. But at some point, they start to match, and then what we are seeing here is really that the recipient trait can check the evolution of the landscape, the development of the landscape very closely. And the right is, the plot on the right is showing the discrepancy of these two variables. Okay, so this is the recipient trait, the evolution of the recipient trait. It is nothing surprising here. Now let's look at the evolution of the geomorphic trait, which decides the capacity for the plant to modify the environment. This is what the evolution of the geomorphic trait look like. The one on the left, on the x-axis is the geomorphic trait value. The one on the y-axis is the recipient trait value. You can look at this as two stages. First, in the early stage of the landscape development, it is positive niche construction because the landscape elevation is low, and the plant actually, the preferred elevation by the plants is actually higher than the actual elevation. So if the plant can do a lot of niche construction activities, then those plants are going to be preferred and their frequency is going to increase in the population, right? So then you will see the value, the population mean value of the geomorphic trait start to increase in this stage, which I call this as the positive niche construction phase. But as the landscape evolved to be at a higher elevation state, then at that point the plant can't catch up with the elevation anymore. So at that stage, you would assume that is plants that has lower capacity to modify the environment are going to win. And so at the population scale, the population mean value of the niche construction trait is actually going to move to a smaller value. So this seems to be fine, meaning that the niche construction trait is actually evolving directionally towards an adaptation direction, and natural selection is working. Except one interesting thing here, that you notice that the stochasticity associated with the niche construction trait is really high. What I mean by stochasticity is represented here by these standard deviation, represented by these blue bars. And the stochasticity associated with the recipient trait, which is this red bars, is a lot smaller than these blue bars. So there's a lot of stochasticity associated with the evolution of the niche construction trait. In fact, if I change some of the parameters in the model, in particular, if I change the dispersal strategy from local dispersal to long distance dispersal, and if I increase the lifespan of the plants to like 30 years, from 30 years to like 500 years, I see that the niche construction trait stops evolving. What you are seeing here on the most left is the plant population with local dispersal and short lifespan. And this is the plot that you've already seen in the previous slide. It actually evolved. The niche construction trait can evolve. But now in the last three plots, I either increase the dispersal or increase the lifespan. You see that on the x-axis is this geomorphic trait. It basically it does not evolve anymore. It is overwhelmed by the stochasticity associated with evolution of the niche construction trait. So this is some summary statistics. Let's look at the spatial pattern of these two traits, how they evolve in the landscape. And let's just look at the model. I run the model for 100,000 years. This is a 5,000 year snapshot, 5,000 year snapshot of that time scale. There's an emergence of advantageous recipient trait emerging at this time step. And you see that over the next 5,000 years, this recipient traits quickly take over the whole population and spread across the landscape. Because it has some advantage, and then this will spread across the population. There's nothing surprising. But think about it, how simultaneously, how would the geomorphic trait evolve within this 5,000 time window? And here's the result. This is what is the evolution of the geomorphic trait look like within the same time period. You don't see a lot of association between this recipient trait and the geomorphic trait. And in fact, you can quantify the statistical correlation between these two traits. And first, overall, you see that the correlation coefficient of these two traits is very low. And another thing is that I manipulated the feedback strengths, the strengths of the geoevolutionary feedback. And if I make that feedback stronger, I can actually increase this, this coupling between these two traits to some degree, but not a lot. Okay, so this is some key result for the evolution of the traits. Now let's look at the implication of letting the plants evolve over time. How would that change the landscape geomorphology? So this is the second part of the result. So here the different plots are different stages of the landscape development. What you are seeing here is when I turned off the plant evolution, so only the physical process operating in the model. This is what landscape look like at different stages of the development. Now, let me turn on the evolution of the plant. This is what they look like. If I let the plant evolve over this 100,000 year time scale, the landscape look completely different if the biological evolution of the plant is not accounted for. There's these different landscapes, and the difference why they look different is because this result from 3 models where each model has different strengths of this geo-evolutionary feedback. So I can tune like how closely the feedback, like these two things are coupled. But the key message here is that they look very different. Now, another thing that's really interesting is that it also these different landscapes also completely change the evolutionary trajectory for the geomorphic traits. So this is the one where I have this is 3 models with 3 levels of the strength of the geomorphic feedback. This one, you see that this is a low relief, and this is corresponding to this one, that we see that the geomorphic trait can evolve directionally and natural selection can work. But if you look at these 2 patterns, we basically see that the—
Xiaoli, I'm sorry, we're at 19 minutes and 30 seconds, so we'll need to start moving on to the next speaker. Sorry.
Okay, so it can't evolve. That's basically my last slide. Sorry about that.
Thank you very much. That was a very wonderful talk.
Thank you. Sorry.
I apologize for the interruption.
No, I apologize.
All right. Thank you, Xiaoli. It looks like we don't have time for questions, so we can save those for Slack or for the Zoom chat. We'll now be moving on to William Godsoe, who will be talking about disentangling niche theory and beta diversity change. William, I'm not getting sound from you. Are you muted?
Naturally. Any other technical glitches we'd like to sort out while we're at it?
Everything else looks great.
Perfect. Okay. I was just going to say thank you very much for having me here. I live in New Zealand and often feel isolated. Sometimes that's really good, but it's really lovely to get to talk with people in other parts of the world right now. I'm going to be talking about disentangling niche theory and beta diversity change. So myself, and I think many other people here, are really concerned about changes in biodiversity. We really care about especially how people, exotic species, climate change are going to change the distribution and abundance of species over planet Earth. One of the pieces of biodiversity that is particularly important, but particularly difficult to study, is what I'm going to call beta diversity. So this is differences among ecological communities. I've put a schematic of this up here, and you'll be seeing a lot of this throughout the rest of the talk because pictures are better than equations as much as I can do. So we've got 2 communities, one ellipse and another ellipse. This one has all blue species, all of the blue species. This one has all of the gray species. And so this is high beta diversity, where each location, the 2 ellipses, has different set of species. And this corresponds to what we might expect in a natural environment where you have different locations with distinct ecological communities. We're really worried about a phenomenon I'm going to call biotic homogenization, which means that in the past, we might have had high beta diversity. And as we move to the present, we're getting to landscapes where every location looks roughly the same as every other. Pictorially, this might look something like this, where in the past, each community is separate, has different species, but in the present, all communities look the same. We'd like to be able to predict when we'll get biotic homogenization, but oftentimes that's surprisingly difficult. There's a nice review by Secola et al. A couple of years ago that showed that many of the times that we'd expect to see biotic homogenization, we'd get the reverse. And so we'd like to be able to make better predictions. And I'm going to try to build up a framework in this talk that lets us do that. A lot of the predictions that we have about biotic homogenization right now focus on what I'm going to call niche theory. And so the idea here is that we look at the environmental requirements of different species in different locations and try to go from there to build up to an understanding of biotic homogenization. So for example, in this photo, this is the Banks Peninsula, which is about an hour away from my house. You've got gorse here, the yellow flowers, which is an exotic species that was introduced to New Zealand from the UK. It's a species with a fairly broad niche. Many environments are suitable to gorse in New Zealand. And so based on niche theory, we might just say, oh yeah, the species is going to lead to biotic homogenization as it becomes more abundant in New Zealand. So that's the way our predictions work. But I'm going to try to dig into this and show that I think these predictions we have are not as useful as we'd like. To do this, I'm going to take this scenario of an exotic species and try to strip it down to its basic parts. So here I've set up a very simple version of the invasion of a species. I've got two communities. One with a habitat specialist, so this species can only live in this community. And then I've got a newly arrived generalist invader. So it's invaded one habitat patch, it can grow in this patch, it can disperse to this patch, and it can grow in this patch. So the invasion of a habitat generalist, this is supposed to lead to biotic homogenization. And I'm just going to show you what happens when you do a simulation of this with logistic growth. And I have to remember, this is the way to turn the button here. So here's a simulation of this scenario with logistic growth. The gray species is abundant in the one patch that it occurs in. The blue species starts out in one patch and it grows. As it disperses to the other patch, it grows there as well. That is supposed to lead to biotic homogenization. But if you actually measure beta diversity, what you find is the reverse. So beta diversity shoots up for the first part of the simulation before gradually declining to close to where it was before. And so this is going to be my motivating example that maybe we can do better at predicting beta diversity if we think a little bit more carefully about what's going on. In the talk, I'm going to try to do three things to help us to think more clearly about beta diversity. I'm first going to identify individual species contributions to beta diversity. I'm then going to identify individual species contributions to biotic homogenization using a technique from evolution known as the price equation. And then I'm going to show how these contributions can be measured in nature using an empirical data set from the Blue Mountains of Jamaica. Okay, so the first part of this talk is going to be to think about beta diversity not as the contributions of different communities, so this community versus this one, but of individual species. For reasons that are more than I want to talk about in a talk right now, it turns out that there's a really nice measure of beta diversity to start thinking about these issues, and that's the additive partition of Shannon-Weiner diversity or Shannon-Weiner entropy. And in Slack, I'm more than happy to geek out about why this is later. I've done a couple of papers, either myself or collaboratively, that clarifies why this is a nice metric to start with, and you can use it to move to other metrics later on. Okay, so for this metric, what you're basically doing is comparing rarity scores for your species in different regions. So I've got my ecological community here. Sorry, my two ecological communities here. I can start by measuring the overall rarity of my species across both communities. So half of my individuals are blue and half of my individuals are gray. And so the rarity score is based on this. It turns out that it's a logarithm, but we don't like logarithms, so I won't talk about that. We're just putting it in the picture for now. So that's overall rarity. We can then think about the rarity of each species within each community. So in this community, the blue species is rare, and in this community, the gray species is rare. Again, this is measured with logarithms, but I've illustrated this pictorially here. So the blue tree is larger here because negative log pi given j, the probability of species I given that you're in patch j, is big, and it's small for these ones over here. So we've got separate rarity scores for each individual in our community. What beta diversity is measuring is a difference in those rarity scores, so that this tree is rare in its community relative to how rare the blue tree species is across the meta community. This tree is more common relative to its score across the entire community, as is this one. So that's the contributions that individuals make to beta diversity. The contribution of a species is then the average of the contribution of all individuals. So you take one individual 2 individuals, 3 individuals, and add up each of the scores that they have. Okay, so then that's the contribution that an individual species makes. I'm going to call this Zi beta, and you'll see this throughout the rest of the talk. Beta diversity is going to be a way to add up the contributions of each individual species, weighted by the frequency of that species or their relative abundance. And biotic homogenization is the change between one time step and another of beta diversity. So this is sort of the setup for the entire, for describing biotic homogenization. It's an equation that we don't work with all the time in ecology, but it turns out that evolutionary biologists have found that a very insightful way to analyze change in equations like this is to use what's called the price equation. And basically what that does is it breaks down overall change into the contributions of 2 mechanisms. So you've got one mechanism which is describing selection among species on Zi beta, and a second mechanism which is describing change in Zi beta. Pictorially, in equation form, it's a little bit hard to see what's happening, but there's a nice pictorial interpretation of this. So this is selection on Zi beta. We have 2 species in the past. One of the species is really successful. So the gray one produces 2 descendants, whereas individuals of the blue species produces a single descendant. So the gray one succeeds. In the past, this species was evenly distributed. So it has a low Zi beta score, and that leads to biotic homogenization. In the present, the communities are more even than they were in the past. So that's selection on Zi beta. This is simple change in Zi beta. The key idea here is that across the meta community, neither species does better than the other. So in the past, half the individuals are blue and half are gray. In the present, half the individuals are blue and half are gray. But even though there's no overall advantage to either species, you still get a change in beta diversity, and you still get biotic homogenization because each species does well in one patch and does poorly in another patch. Okay, and so to sum up what this says for Shannon-Weiner entropy, a species contribution to beta diversity depends on this relative rarity score. A species contribution to biotic homogenization depends on the link between the success of that species, both overall and in particular patches, and these relative rarity scores. And so what I've also tried to show is that using the price equation, we can separate these 2 contributions to better understand what drives biotic homogenization in nature. And once we have measurements of biotic homogenization, so we measure beta diversity at 2 different time steps, it's relatively easy to tease apart the contributions of these 2 mechanisms. As an example of this, I was able to get data from my colleague, Peter Bellingham, who had done some collaborative work with Edmund Tanner, setting up long-term plots in the Blue Mountains of Jamaica. It was one of those things where you see the talk and go, oh, that's amazing. I really want to grab that entire data set. So they had a series of 4 plots set up in the Blue Mountains of Jamaica. This is rainforest habitat. And their study was initially set up to measure differences between habitat specialists and habitat generalists. And they did this by comparing 3 nutrient-rich sites to 1 nutrient-poor site. So this is a nutrient-poor site, which was called Moore. And at that site, you had a bunch of habitat specialists that succeeded. So you've got this study designed to set up beta diversity. They started doing censuses in the 1970s. In the 1980s, the site was hit by Hurricane Gilbert. So it's a very large hurricane that came through. And they were able to try to measure Shannon-Weiner diversity across multiple time steps between the 1970s and the present. And for the next part, I'm going to show you what happened in the period that I think is most interesting, which is between 1984 and 1994. So this is the period before Hurricane Gilbert hit their site versus after after Hurricane Gilbert hit the site. In their original study, they said that in Tanner and Bellingham 2006, so this is one of the papers based on this work, they said that there was evidence of greater success for habitat specialists, and we can see if that makes sense based on sort of a formal analysis of biotic homogenization. We can start by looking at which species contributed most to beta diversity. So this is which species had high Zi beta scores. And consistent with what we described before, habitat generalists had low Zi beta scores. So this is a species here that occurs in all 4 sites. The purple lines describe the frequency of that species in each of the 4 sites. The blue lines show what that frequency would be if the species is evenly distributed across all 4 sites. So this species is widely distributed, it does well in all sites, it has a low Zi beta score. This species is a habitat specialist, and so it has a high Zi beta score. And you can see that in the Moore site, it's extremely frequent. And in the other 3 sites, it's infrequent. So sometimes this Zi beta score connects nicely with what we think about as standard niche differences, but sometimes it doesn't. And we can see this here. So this species here, Cinnamonum montanum, is a species that's just rare. They censused all plants in the plots, and 2 of those plants total belong to Cinnamonum montanum. And you might speculate, well, maybe it's a hyper rare specialist in the Blue Mountains of Jamaica. That's a little bit hard to tell because it's a difficult place to access and study. There's a very good chance that this is just a species that's rare. And there are 2 individuals, and by luck, they both happen to be in the same site. That still leads to a high Zi beta score. So the contribution to beta diversity doesn't care about whether it's a specialist or rare. So once we've identified which species contributed a lot, we can then check to see whether species that contributed a lot to beta diversity succeeded relative to others. And the answer is no, they didn't. So in this time period, there was no selection on Zi beta. Didn't matter what your Zi beta score was, you were just as likely to leave descendants as any other species, and relative rarity scores stayed roughly the same. The only difference in the data set, and it's one that we're able to sort of pull out exactly, is that you had a small number of individuals belonging to new species that made it into the community, like this exotic species here, which is a species of Pittosporum, which had come in soon after the hurricane came through. And so that made a modest change to beta diversity, but overall there was a very limited effect, though other diversity components changed a bit more. And so this is most of what I wanted to show. So biotic homogenization occurs when species with low Zi beta scores succeed, or when Zi beta scores decrease. Niche characteristics are sometimes useful, but they will mislead when they fail to predict the Zi beta scores. And in the Blue Mountains, we can measure the contributions of each species to beta diversity, but Zi beta scores did not predict species success. All right, that's it. Thank you very much for listening. I'm ready for questions. Yay.
People always need some time for typing at least.
Yeah. Well, if people are just going to wait, I put in some pre-cooked questions. I'll see if I can pull those slides up, and then if someone types in, just feel free to interrupt.
Yeah, go for it.
So one of the things that people often ask is how this applies to other, yes, this is that slide. So can this be used to other diversity metrics like hill numbers? So hill numbers are a framework that's set up by Jost and MacArthur and a couple of others that convert different diversity metrics into common units. Can this be applied to that? So if we take the hill number associated with Shannon-Weiner entropy, it's really easy. And in fact, this is just the slide I was going to get to right here. So this is the change I described for additive Shannon-Weiner entropy. To convert it to hill numbers, you get this. So then you just raise the equation to E on both sides. That changes the interpretation a little bit. So instead of looking at change in beta diversity, so like the difference between beta diversity in one time step and another, you're looking at the ratio of beta diversity between one time step and another. So the interpretation is a little bit different. You have to think in terms of ease, which is hard to do in a talk. So I skipped that here. Sorry, so I didn't put it in the main talk there, but it works really well. The other thing about this is you can generalize to other Hill numbers, but I had to email Steve Frank, who's one of the experts on the price equation, who generalized the price equation for that. And so Frank wrote a paper with me as a co-author. He did most of the work, which I want to clarify, that shows how you can apply this to other Hill numbers. There was another question, but I don't think I have time here. Is that right?
Really quick. I mean, there's like, the question was from Inbar, how do you think we can resolve the issue of rareness versus sampling effort?
Okay, so I think there's more work that has to be done on that. Chow, and I forget the other author, recently showed how to correct the Shannon-Weiner entropy metric because it has this bias due to poor sampling effort. And I believe that that correction could be applied to this, but I haven't checked that for sure. So I think that was part of what was so great about the Tanner and Bellingham data set, is that they did a complete census of all the plants within their plots. And I'm trying to get this first part of what works in theory sorted out before being able to deal with some of the other statistical issues that can come up.
Thank you very much, William, for a great talk. Next up, we have a pre-recorded talk by Fernanda Caron on do geographic range sizes evolve faster in endotherms?
Hello, everyone. Thank you for coming to my presentation. My name is Fernanda Caron, and I'm an undergraduate student at the Universidade Federal do Paraná in Brazil. Today, I'm going to talk about a study entitled Do Geographic Range Sizes Evolve Faster in Endotherms, which was carried out in collaboration with my lab mate, Raquel de Viezo, and my advisor, Dr. Omar Soufi. One could say that the geographical distribution is possibly the most important predictor of the evolutionary fate of a species. There are several ecological and evolutionary processes that can change these distributions, including allopatral speciation and local extinction, but relatively little has been achieved in terms of general principles of geographical range evolution. One of the recent discoveries in this matter is that different range limits can evolve in qualitatively different ways. For instance, let's define the low latitude limit of a geographical distribution as the latitude of a given geographical distribution that is closest to the equator. Likewise, the high latitude limit refers to the latitude closest to the poles. So what P and Meyer in 2017 did was to use phylogenetic comparative methods to test whether low and high latitude limits evolve at different rates. And they tested this using a broad sample of terrestrial vertebrates, focusing on mammals and squamates. The rationale for this test is simple. The mechanisms underlying low latitude limits might include tolerance to high temperatures and exposure to desiccation, whereas high latitude limits could involve mechanisms such as tolerance to low temperatures. Given that these mechanisms involve different physiological processes, one could imagine that they might not share the same evolutionary rates. And indeed, what was they found? The y-axis indicates the estimated evolutionary rates going from low latitude, centroid, and high latitude. Along the x-axis, we see the different mammal groups, Artiodactyla, Carnivora, Chiroptera, Marsupialia, and primates, and different squamate groups, Endimorpha, Geckoda, Iguania, Lacertoidea, Syncoidea, and snakes. We see two main results from these graphs. First, High latitude limits in terrestrial vertebrates invariably evolve faster than low latitude limits. This difference was substantial, with high latitude limits evolving 1.6 to 4 times faster. These consistent differences are intriguing, particularly given the obvious ecological differences between these taxa. The second issue, which was actually not explicitly addressed by P. and Meyer, is that the rates in mammals are substantially higher than squamates, which can be seen by looking at the scale in the y-axis. This difference may suggest that possibly the evolution of homeothermy may influence the evolution of the geographical ranges, possibly by affecting the evolution of their climatic niches. This study of Roland and collaborators actually found evidence for this idea, with climatic niches in 2 endotherm groups, birds and mammals, evolving their climatic niches faster than in ectotherms, that is, squamates and amphibians. Given this evidence to date, one might wonder how specifically endothermy could actually affect the evolution of range sizes. Therefore, if we think of endothermy as the internal buffering of body temperature, this could allow for species to be less constrained physiologically. Endotherms are characterized by higher metabolic rates, as Clark and Portner have shown in their study. These rates might lead individuals to forage farther and space themselves more widely to meet their energetic demands. As a consequence, they would end up having low population densities, larger home ranges, and finally larger species distributions to maintain minimum viable population sizes to avoid extinction. Therefore, endotherms might be able to attain broader limits in their ranges, resulting in relatively larger range sizes. So what we wanted to test in this study was the extent to which endotherms are characterized by qualitatively different evolutionary dynamics with respect to 3 criteria. First, if they have larger geographical range sizes when compared to ectotherms. Second, if they show faster rates of range size evolution. And third, if they present faster changes in the centroid of geographical ranges, that is, their latitudinal position. We explored these hypotheses with a large dataset of terrestrial vertebrates, more specifically mammals, birds, squamates, and amphibians. Basically, what we did was to compile the range sizes and phylogenetic information for all of these taxa, and then analyzed how their ranges are distributed across different subclades. We choose to focus only on these subclades because they are more ecologically homogeneous, which facilitates the interpretation of the analysis. So, to better understand our results, here is a representation of the types of histograms I'm going to show you. On the x-axis, we have the log10 transformed areas of each species, and on the y-axis, the frequency distribution of range sizes. To facilitate visualization, we added these great lines to each graph for comparison. So the solid line indicates the area of Europe, the dashed line indicates the area of South America, and the dotted line the area of Africa. What this means is that ranges cannot be much larger than the areas of those continents, and this explains the truncation we have when analyzing the distribution of all species. And here it is, the range size distribution for all terrestrial vertebrates that we compile. This is a typical range size distribution found in other taxa as well, which is almost log normal with a left skew. This red line indicates the mean of the distribution. And the reason why we used this color palette was to make it easier to see the differences between subclades, which will become clear later. When we compare all subclades, we see some interesting patterns. For instance, highlighted in here, we have squamates. And we see that they tended to show lower average range sizes, with a substantial proportion of small ranged species, as indicated here in these darker colors. For amphibians, we see basically the same patterns, with a greater frequency of small ranges and a lower average range sizes. However, when we look at mammals, we see some interesting differences. First, the relative frequency of smaller ranges is substantially lower, with a higher frequency of large ranges, which can be noted by the change in color distribution. Besides, the average range sizes are also higher when compared to the ectotherms. And these patterns also appear in birds. An important thing to be noticed is that taxa that are good dispersers, such as Coradriforms and Chiroptera, tend to have more right-skewed distributions in relation to other taxa with relatively lower dispersal capacity, such as Piscitaciforms and Diprotodontia. In addition, one curious exception to this pattern between endotherms and ectotherms are snakes, which not only have larger ranges than ectotherms in general, but also have relatively fewer small ranged species. Interestingly, this pattern is not constant among all snakes, with Columbroidea showing a more typical ectotherm range size distribution, whereas viperids and particularly elapids show a clear decrease in the frequency of small ranges, characteristic of many endotherm range distributions. However, we still don't know why we have these differences in snakes, and current research in our group is trying to figure out these differences. We also fitted a model of Brownian motion, both in the range sizes and the latitudinal position of the subclades. What we found was that generally endotherms, highlighted here in red, tended to have the highest rates of range size evolution, with all but 2 of the 11 highest rates, while 6 of the lowest rates were found in ectotherms, highlighted in blue. The main exception for this, again, was snakes, which have rates comparable to an endotherm. Likewise, endotherms also show faster changes in the centroid of geographic ranges. Interestingly, 9 of the highest rates were found among endotherms, while 8 of the lowest rates were found among ectotherms. And the only exception, again, was snakes, which showed higher rates than Diprotodontia. And as we had 1,000 alternative topologies available for all taxa, we repeated all analysis for each of them to make the results robust to phylogenetic uncertainty. And here we have the 95% quantiles of the results. So why do endotherms seem to have a faster tempo of evolution? One of the potential explanations for these differences involve metabolism. Endothermic vertebrates might require at least an order of magnitude more energy than ectotherms. For instance, food metabolic rates are either stable or decrease slightly with temperatures for endotherms, whereas they generally increase for ectotherms. This could lead to opposing latitudinal gradients of expected field metabolic rates. If these patterns are indeed general for the field metabolic rates of the taxa we study, they can potentially translate into distinct geographical variation in their metabolic budgets. In 2013, Agosta and collaborators explored 3 main hypotheses relating metabolic rates and range size in mammals, which might provide us with some clues to explain the results. First, the thermoplasticity hypothesis that says that higher basal metabolic rate would be associated with increased thermal tolerance, thus allowing for larger range sizes. In other words, the increased thermal buffering provided by the high metabolic rate would allow for species to have broader distributions outside of what would be their ideal temperature if they were ectotherms. Second, the activity levels or dispersal hypothesis, which says that higher basal metabolic rate would increase activity levels and dispersal potential of species, leading to increases in potential range sizes. In other words, the improved dispersal capacity provided by the higher metabolic rates would directly improve dispersal rather than simply the thermal tolerance indicated in the thermal plasticity hypothesis. Finally, the energy constraint hypothesis, which posed that the elevated energy requirements associated with high basal metabolic rate would constrain individuals to forage farther and space themselves more broadly, leading to ultimately larger geographical distributions. All of these hypotheses might play a role in explaining our results, and teasing apart their relative contributions might provide valuable insight into how the evolution of homeothermy changed the way geographical ranges evolve in these taxa. So the final take-home messages that we can obtain from our research is that there are evidence for all hypotheses tested. That is, endotherms indeed have larger geographical ranges, also have faster rates of range size evolution, and faster changes in the geographical positions in their ranges. Thank you for attending to this talk, and I'll be happy to take any questions.
Fernanda is online with us if anyone has questions.
As usual, if there are any questions coming in later, I'll put them in the chat, Fernanda, so you can go check them if something comes up.
Yeah, okay.
I'll chip in with a question. This is Dan Bolnick. Very nice talk. Nice to see this work. One question that I always have with range size comparisons is about taxonomic practice and whether differences in range size can ever be due to how taxonomists in different working on different groups choose to split or lump. And so, you know, if there's a culture of subdividing species more finely, geographically, you get more species with smaller ranges. If we tend to lump, we get fewer species with larger ranges. And that's probably not contributing to this situation, but I'm just curious if you can comment on if you've thought at all about that issue, because I've never really figured out how to address that effectively.
Yeah, we were working on this paper with the shapes from IUCN, and we have basically the same problem with taxonomy. So if there are errors in taxonomy, we have errors in the range sizes too. But we believe that with works in this scale with thousands of species, there aren't a specific way we can resolve it. So we believe that these errors wouldn't bias or result in any way. So that is, we have to start from a point. So.
Right. Excellent. Well, thank you. Very nice.
Yeah.
All right, we had a few more questions coming in. One from Juliano who says, great work. What do you think the patterns will apply to other groups, for instance, insects?
Yeah, I'm not sure how to respond to it. I don't have much experience with insects. And we have the reason why we haven't tested with other groups is the fact that we don't have much data on the distributions of other groups. So I don't know, but I can look for the answer and give back to you.
We have another question from Eva Fisher who says, like, sorry if I missed this, but did you control for variables like body size or metabolic rates in these comparisons? Perhaps related, is there evidence for these patterns holding within endotherms based on differences in metabolic rate and body size?
Yeah, we think of added body size to our analysis, but the reason why we didn't add it is because we don't have much data on body size on all the species that we were working on, more than 20,000 species. So that it is one of the reasons, and the other reason is that if we added one more trait to the analysis, it would become much more complex, and we didn't want that, so we didn't want we want a first approximation to the problem. So that is why we didn't add it.
Thank you very much, Fernanda. If you have any more questions for Fernanda, please post those to the chat or to the Slack channel. Our next pre-recorded talk will be from Haley Hanson on epigenetic potential in house sparrow, Passer domesticus, introductions.
Hi everyone, thank you for coming to my talk. My name is Haley Hanson and I'm currently a PhD student at the University of South Florida. I'm going to be sharing some of my dissertation work today on epigenetic potential in an introduced passerine. So introduced species are among the greatest threats to native ecosystems, and they also cost billions of dollars a year to manage. However, they also present opportunities for understanding important evolutionary processes and other phenomena, such as how organisms adapt to new environments. So we can turn to some of the most successful introduced species, such as the house sparrow, for insight. To illustrate their overwhelming success, we can watch their expansion across multiple continents starting in the early 1800s. Here, the green area represents their native range, and the yellow area represents their introduced range. So in less than 200 years, they've expanded both their native and introduced ranges considerably. And in each of these new areas, they've most likely faced new challenges, which necessitates different traits and responses to deal with these. So how was the species able to overcome all of these challenges? We expect the answer lies in epigenetic mechanisms, which can help individuals overcome genetic challenges such as bottlenecks or founder effects. In one of their most recent introductions into Kenya, populations with low genetic diversity, as seen here on the x-axis, often have high epigenetic diversity, as seen here on the y-axis, meaning they might compensate for low genetic diversity with high epigenetic diversity. And this epigenetic diversity may allow for more variation in phenotype than from genetic differentiation alone. We hypothesize that individuals may differ in their capacity for epigenetic mechanisms to work within the genome and contribute to phenotypic variation. We've termed this epigenetic potential. And stated a little bit differently, individuals may differ in their capacity for environmentally induced phenotypic plasticity through epigenetic mechanisms. We can think about epigenetic potential from the viewpoint of one epigenetic mechanism, DNA methylation. DNA methylation occurs when methyl groups are added to DNA, generally at CpG sites or cytosines followed by guanines in the sequence. DNA methylation can be induced by environmental conditions and can suppress or enhance gene expression depending on where the CpG site is located. So most studies have focused on CpG sites in the promoter region of genes, and this is generally correlated with the suppression of gene expression. I really like to focus on the word capacity here and think about how this can happen. So when we think about DNA methylation in terms of epigenetic potential, one form of this could be in the number of CpG sites a gene has. I want to reiterate that this form of epigenetic potential is occurring at the genomic level. And as an analogy, I'd like to picture two stereosystems. The first has only one knob for adjusting the volume, and the second has multiple knobs. These can be adjusting for volume, bass, treble, balance, etc. So while both stereos produce sound, so they're both functional, the second system with more knobs allows for finer acoustic tuning. So in some situations, this is not going to be needed, but the knob still exists, so when the relevant occasion arises, they can be used. If we think about those knobs as CPG sites that can be methylated or not, the individual with more CPG sites or higher epigenetic potential might be better able to match their phenotype to the environment by regulating gene expression more precisely. Consequently, epigenetic potential may be influential for success at range edges and in introductions, especially when it resides in genes important for coping with novel challenges. So to understand how epigenetic potential might be working within the house sparrow system, we looked at a candidate gene that's been found to be important to range expansions previously, the toll-like receptors. The toll-like receptors, or the TLRs, are considered some of the first lines of defenses against generalist parasites, as they recognize evolutionarily conserved components of these pathogens. So, for example, toll-like receptor 4, or TLR4, encodes a protein that detects for lipopolysaccharide, or LPS, which is found in gram-negative bacteria. So once this LPS is detected, pro-inflammatory cytokines are activated and an immune response begins. Within a range expansion, this might be important for individuals that are encountering generalist parasites or parasites they don't have an evolutionary history with. So for my first study, we wanted to know whether epigenetic potential differed between 3 native and 8 introduced populations of house sparrows. So we asked about the number of CPG sites or the level of epigenetic potential in the TLR4 promoter. We would expect that epigenetic potential is highest in these introduced populations where they might be coping with these unfamiliar parasites, and because this might allow gene expression to be regulated more precisely. And we did find that on average, introduced populations maintained higher numbers of CPG sites than native ones did. So again, the introduced population is represented here in yellow, and as you can see, they have more CPG sites than their native counterparts. For my next experiment, we really wanted to delve into the functionality of epigenetic potential because we've made these predictions about the effect on gene expression. So for this study, we caught birds in the Tampa Bay area that differed in epigenetic potential. And then we injected them with either lipopolysaccharide, LPS, the TLR4 agonist, or phosphate buffered saline, PBS, as a control. From all of the birds, we took blood samples at the time of injection, 4 hours post the injection, and 6 hours post-injection. And we expected that in birds injected with LPS, we would see TLR4 expression increase over time, peaking at 4 hours, and then decreasing around 6 hours. And we also expected that birds with high epigenetic potential that were exposed to LPS would show both greater inducibility and reversibility. So basically a greater peak in expression, but also a better ability to return to baseline levels of expression. In addition to blood samples, we also took samples of the liver and the spleen at the end of the experiment. We expected that epigenetic potential might relate to tissue-specific gene expression. As epigenetic potential is encoded in the genome, this might allow for differential regulation across scales from single cells all the way up to whole tissues. Both the liver and the spleen play an important role in immune responses to bacterial infection, but as we only sampled these organs at one time point at the end of the experiment, we couldn't make explicit predictions about the directionality of TLR4 expression. We just hypothesized that it might differ between types of samples. So in the blood, we found that high epigenetic potential individuals, or those with more CPG sites, which are represented here in the dark green, we found that they had higher expression compared to their low epigenetic potential counterparts seen here in light green. We measured this by taking the area under the time curve here. Surprisingly, this relationship was independent of LPS, and we found a pretty weak effect of LPS overall. It was much weaker than we had originally anticipated. Just to view these results a little bit differently, I've plotted area under the curve values for each individual here. And you'll notice that high epigenetic potential individuals had almost double the amount of toll-like receptor 4 expression than low epigenetic potential individuals did. So this was a pretty strong effect here. Interestingly, we found that epigenetic potential was associated with TLR4 expression in each tissue that we looked at. So the blood, as you saw on the previous slide, liver here in the center, and the spleen here on the right. And while these differences appear small, the effect was significant. I also want to point out that the directionality was not the same between these types of samples. High epigenetic potential individuals had lower expression in the liver and in the spleen compared to low epigenetic potential individuals. And this trend was reversed in the blood, as we saw previously. Lastly, we found a really interesting and surprising relationship between epigenetic potential and sex. We found this in each of the tissues we examined, but for the sake of time, I'm just going to show the blood data here. And you can see that high epigenetic potential females, shown here in red, exhibited both high inducibility and reversibility of expression. So they were able to mount a response by 4 hours and return all the way to baseline levels of expression by the end of the experiment, while low epigenetic potential females, shown here in pink, exhibited this same trend, but it was lower in magnitude. And you can see that as their expression in the last sampling period at 6 hours was not different from either baseline or induced levels. And in males, we found that both high and low epigenetic potential groups showed inducibility, but neither group was able to return to baseline levels of TLR4 expression, so neither were exhibiting reversibility. So overall, high epigenetic potential females show evidence of higher inducibility and reversibility of TLR4 expression. And I just want to reiterate that these results are independent of treatment with LPS. So to summarize, introduced populations of house sparrows have higher epigenetic potential than their native counterparts do. And epigenetic potential is associated with TLR4 expression across the blood, liver, and the spleen. Lastly, high epigenetic potential is associated with higher inducibility of TLR4 expression in the blood, but only females exhibited reversibility. So we see these interesting relationships between epigenetic potential and introductions and epigenetic potential and expression in toll-like receptor 4, but we next wanted to ask if these trends were more generalizable. Could we detect a difference in epigenetic potential across a recent range expansion while looking across the genome? For this study, we sampled 5 cities across Kenya, and here we used the distance from Mombasa, or the site of initial introduction, into Kenya as a proxy for population age. This measure represents how much time the house sparrows have persisted in each of these areas. Using this metric, we asked how CPG sites differed between Kenyan cities after double digest RAD sequencing. This form of sequencing returns a random subset of the genome based on restriction sites. And lastly, we asked whether patterns of epigenetic potential arose via selection by comparing values of Tajima's D and Fei-Nwu's H across the range expansion between CPG sites and non-CPG sites. And across the range expansion, we expected more CPG sites towards the range edge or higher epigenetic potential in these younger populations. We predict this might occur through selection for CPG sites at the range edge, but not at the range core. Negative values of Tajima's D suggest positive selection or population expansion, but using only this value would not let us distinguish between these alternatives, so we can also use Fei-Nwu's H to help distinguish between these. If selection is occurring for CPG sites, we expect to see negative values of both Tajima's D and Fei-Nwu's H. And we also think that we might see a decrease in these metrics across the range expansion towards the range edge. We hypothesize that as new areas are colonized, epigenetic potential might be selectively advantageous early, but eventually replaced by genetically canalized responses as populations adapt to local conditions. To walk through some of my results, on the x-axis, you'll see distance from the site of introduction, or Mombasa. That is represented by 0. That's the oldest population. And 885 represents the range edge population, or the youngest population. And the y-axis, you'll have a relativized measure of CPG sites. So we found that epigenetic potential was highest at the range edge and decreased towards the range core, which was similar to what we originally predicted. This trend was very strong, and again, we expect that this might be beneficial for those range edge populations to cope with the challenges that they face expanding into new areas. Next, we found that values of Tsujima's D significantly declined towards the range edge for CPG sites, represented here by the green line, but not for non-CPG sites, represented by the blue line. And this was also in line with our initial predictions. When we looked at Fei and Wu's H, we did not see the same trends as we did for Tsujima's D. Neither CPG sites nor non-CPG sites exhibit a significant change across the range expansion for Fei and Wu's H. However, I do want to point out that we see extremely negative values of Fei and Wu's H at the range edge compared to the range core, which suggests selection for CPG sites at the range edge, but we really cannot completely rule out the other processes that I mentioned, such as population expansion, but also allele surfing and purifying selection occurring at the range core. So to summarize, we revealed that epigenetic potential follows the patterns that we would expect, with birds on the range edge having the most epigenetic potential or the highest number of CPG sites. And as CPG sites are genomic motifs and they can be inherited and selected for, we asked about selection and found that it seems to be acting on CPG sites at the range edge, but we cannot completely rule out other alternatives. I want to reiterate that these trends were detected even though we looked at basically a random subset of the genome. We didn't target specific genes that were important to range expansions, so these are really exciting results for us. Overall, our results suggest that epigenetic potential has been playing a role in house sparrow range expansions. Future studies investigating other range expansions and contexts will be really important in uncovering whether it truly represents a type of adaptive plasticity, and if so, epigenetic potential might be applicable across many fields besides invasion biology. With that, I'd like to thank my funding sources and all of the undergraduate researchers that made this work possible. I'd be happy to answer questions via email.
With that, I'll welcome everyone back to Contributed Papers Section 1 on species distributions in the niche. I'm the moderator. My name is Scott Egan. Our question wrangler is Volker Rudolph, and our tech guru is Mike Whitlock. As moderator, I will announce each speaker, and I will signal at 16 minutes using this little sign, low-tech sign. If you pin my screen as the speaker, you will always be able to see my screen to know that. And then at 19 minutes, I'll also give you a signal. At 19 minutes and 45 seconds, we'll switch screens back to the next speaker. So with that, let's get back to the next speaker. At 40 minutes past the hour, we'll have Abdel Halloway, who will be talking about intraspecific competition can promote ecological specialization. With that, Abdel, you're on mute, but go ahead and get started.
Hey, hi everyone, and thank you for coming to this talk. I guess not physically, but my name is Abdel Halloway. I'm coming to you from Algonquin land, specifically the Wami and Miami, Illinois. And yes, I will be talking about a theoretical model which shows that interspecific competition could promote ecological specialization. So I want to start broadly with this question of specialization. I think like there can be a single question. Our research on ecological specialization can be summed up into a single question. Why would a species evolve a more restricted niche space and utilize few resources? And to my mind, several explanations have been generated. Environmental constancy, there are trade-offs and constraints, adaptation in heterogeneous landscapes, sexual rendezvous, mutualism, coevolution, predation, and interspecific competition. And I specifically want to focus on the interspecific competition and even more so the competition portion. So what is competition's role in specialization? Well, when it comes to interspecific competition, it's believed that when you have interspecific competition between species of different niches, that removes available niche space to an organism or to a species, creating a realized niche. And then over time, the species sort of adapts to become better at the realized niche, and it becomes a new sort of fundamental niche. And so this is all about the availability of the resources, right? With fewer resources available, species better adapts to the resources that are there and become specialized in it. But so we can also flip that, right? What happens when intraspecific competition happens, right? And the idea is that it works opposite of interspecific competition. It's sort of like a mirror image or an enantiomer, right? So if there's more competition for a preferred resource, then individuals gain fitness by using alternative resources, leading to a broadening of the niche and population generalization. This hypothesis is known generally as competitive diversification or adaptive diversification. And it looks something like this, right? If you were to graph it, if you were to look at competition level or population size as a measure of competition level, and the population niche width, as you increase this competition, you go from a more restricted niche width, a very low niche width, to a very high, to a higher niche width. So this hypothesis is largely intuitive, but observational and experimental studies can show a mixed bag. Some show increased specialization with increased competition. Some show decreased specialization with increased competition. And if you look at this review from Jones and Post in 2016, looking at this sort of meta-analysis, they found an overall non-effect, right? So they came up with this broader idea of the intermediate competitive diversification. So at lower levels of competition, there is diversification, there's a broadening of the niche width, but then there's a shrink of greater specialization at higher levels of competition. Basically, as resources are depleted fully, then species move back to consuming the preferred resource. And that leads to a broadly hump-shaped curve, which looks something like this, right? So we have these two hypotheses. So I wanted to explore the idea of resource choice and use under competition. With so many choices, what do you choose to eat? What happens when you face competition? So I and my collaborators created a model of resource use and then varied population size to see how competition changed population specialization. So as an example, let's imagine your resource is popcorn, right? And you're going around, you're eating wonderful Garrett's popcorn, which has the butter and the cheddar and the caramel. Well, actually, let's ignore Garrett's for right now, and let's instead go for Toronto Pop Company, which has all these different flavors. If you want s'mores, you can get it. You want cupcake, you want pina colada, popcorn, it's all there, right? And so you have all these different flavors. And that's an attribute of your resource, right? The flavor of the pop is an attribute of the popcorn. And we can chart the abundance of each flavor. We can now think that maybe there are individuals have different utilization curves, right? So we have, you know, this purple individual, this green, this red, and this blue, right? And they preferably consume different resources. So this green seems to preferably consume the cheddar. This purple preferably consumes the salt and vinegar. And they also have different utilizations, right? So this purple really consumes the salt and vinegar, really goes hard into it, but doesn't really consume anything else. While this blue consumes the strawberry milkshake, but also consumes the caramel popcorn, a little bit of the peri-peri, a little bit of the butter, right? And what they consume is often of what, you know, their own preference, their own utilization curve, but also what resources are available, right? So if this purple individual could really go in hard for the salt and vinegar, it would, but there's just not much salt and vinegar, right? Same thing with this blue individual. This blue individual goes more for the strawberry milkshake and the butter, but doesn't get a lot of benefit because there's not a lot of caramel and, you know, all other ones, right? And this red one in the middle, there's enough resources for it to consume, right? So it gets everything. And then we can imagine, like, now let's add up everybody's utilization curve to get this dotted line, which represents like a total utilization curve of the entire community. And this part becomes important because, you know, if there are limited resources, how do you divide what's available? And we imagine that you can share these resources, you share what's available, right? So, you know, this, both the green, and the red, both go for the cheddar. And because of that competition, they would, I mean, ideally they would like to consume as much as possible, but they have to share it. And therefore that leads to a reduction in what they're able to take. And we imagine this is normalized based upon, you know, this summation, right? The sum of, so this is normalized relative to the total community utilization. So this is our model. And as we do this, just to note some key assumption as we analyze, firstly, we assume there's a single population. So we showed that there's a lot of variation, but right now we're just assuming that there's a single population. And that the utilization curves and the resource curves are Gaussian symmetrical. And what this means is, at least in the model we're using, all individuals have the same preferred resource. So in essence, what you would get is you would get all individuals who are red or individuals who are green. And specifically, because they're symmetrical, they would all be centered around the same preferred resource. We assume some degree of timescale separation so that resources are always at equilibrium relative to the population dynamics. Although we don't really have population dynamics at this point, we're imagining population is a fixed quantity at this point. And then instead of tracking mean individual niche position, we track the mean individual utilization curve width. So we're, instead of tracking, you know, where's this versus this versus this versus this, we're actually asking how wide and how narrow this utilization curve is. So we vary population from a lower to a higher. We fix one parameter and vary another parameter. It doesn't really matter what the parameters are at this point. But then we measure specialization. So sorry, there should be a thing here, but basically red, orange, and yellow are very specialized. They're much more specialized, and the cooler colors, the blues, the purples, the reds, means the population is normalized, I should say. And what do we get? We get this rainbow figure, which I describe as a figure so funky that it would make George Clinton feel proud. And I guess it's hard to describe what's going on. So a better way to represent this is to look at the change from at each increment. As you're slowly increasing the population, does that population become more generalized and more specialized? So here at these lower values, as we're increasing the population size, we're seeing increased specialization. While here we're seeing increased generalization. So the cyan is greater specialization, the magenta is greater generalization. So here at low population sizes, we see increased specialization, while here at high population sizes, we're seeing increased generalization. Here, we're seeing something actually quite like the intermediate competitive diversification hypothesis, where we're seeing increased diversification at low population sizes and increased specialization at high population sizes. And we can do this. So the reason why I said ignore those parameters is we can show this over many different parameters. But it seems like there is a sort of consistency. And we haven't looked at too much. This is very preliminary. That at low population sizes, we see a lot of increased specialization. You know, we see a bit of nuance. So definitely, for example, here it seems to like go up and then down and then back up. So we go from generalization to specialization back to generalization. In some of these areas, we see like, you know, like we see a lot of increased specialization, just pure specialization. We saw some areas where it's, you know, here we don't even see the low population size. So there's a lot of nuance and there's a lot of complexity, but it seems like essentially there's something richer going on here. And we think this is good. So our hypothesis, why we're seeing this phenomenon, back to the idea of the sharing, right? So individuals, so if you are able to consume more of the preferred resource, you know, your resource better than another individual, you will get more of that resource. We think there's actually, that means there's a competitive advantage to specialization, in the sense that if you are more specialized at that on a resource and you're competing with someone, you get more of that resource. And what that means is maybe at low population sizes, that competitive advantage of specialization outweighs the loss that you gain, the loss of giving up alternative resources. And only when competition becomes really high, you know, and then your increased specialization is a lot more marginal, do you see greater generalization. And so the new figure that we present is maybe something a bit like this. That's has a bit more nuance. At these lower values, you see increased specialization due to competitive advantage before seeing more of the classical elements that had been seen before. So going forward, how do we go analyze this for in the future? Well, one, putting it into context, right? Like looking at actual data, looking at actual systems, this would be quite important, you know, and seeing, I think this becomes really important because you can think, well, behavioral versus morphological response, right? So we were talking about, you know, there was no changes in individual niche position, but there were changes in individual niche width. Does one represent a behavioral response or a morphological response, right? If we're looking at these changes in individual niche width, are we seeing much more of a morphological response? Or could it be a behavioral response, you know? Thinking about that, right? Going to, you know, and building upon that, looking at the changes in individual niche position. So we do have data. I did not present it be happy to present it afterwards if people have questions. But looking at the changes in individual niche positions, specifically we look at disruptive versus stabilizing selection. So that gives us an idea of how individual niche position may change. So I ignored the parameters, but we only really looked at 2 parameters. It would be great to look at multiple parameters. We have, I think, 4 parameters in our model that could vary: resource abundance, penalty to specialization, resource spread, and then like intrinsic niche width. They're hard, but just more parameters to see what's, are there other effects? Yeah, only 2 parameters varied. And then thinking about this ecologically, what is relevant, the equilibrium population size. Yeah, it lets us know what's ecologically relevant. So thank you for listening, and are there any questions?
Just a reminder for those with questions, feel free to send them to me in chat, or you can do it through Slack and so I can pass them along.
I guess if there are no questions, maybe we can talk maybe we can take an early break or something like that.
Yeah, we've got 2 minutes until the next talk, so. All right, thank you very much, Abdel. Our next speaker at the top of the hour is Jessica Kowalski, who will be telling us about living on the edge. Habitat microedges increase the dimensionality of species niches.
Perfect, thanks so much, Scott. I just want to make sure that everyone can see my screen because at the beginning, when we practiced sharing screens, it was a little laggy. So any sort of indication would be great. Okay, awesome.
Looks good, looks good.
Beautiful, okay, great. So I just want to thank you all for joining me today. I'm really excited to be here. This is actually my first time both attending and presenting at an ASN conference. I'm just really grateful that we've been able to gather together despite everything that's happening, especially with COVID. So today I'm going to tell you about a project that I worked on just over a year ago that questioned whether habitat boundaries can provide additional niche space when viewed on a micro scale. And I conducted this research while in my undergrad at Western University in London, Ontario, under the supervision of Dr. Hugh Henry. And now I've moved on to work on my master's at the Department of Zoology in the University of British Columbia, under the supervision of Dr. Rachel Germain at the Biodiversity Research Center. So just to start, I want to begin with a reminder of probably the most popular conceptualization of the niche, which is Hutchinson's n-dimensional hypervolume, where different axes describing different resources or different environmental gradients all intersect to ultimately describe a species' fundamental niche space. And it's important to remember that biotic interactions, such as facilitation or competition, can mediate the size and shape of the niche space that a species is able to occupy. So niche theory is often invoked to explain diversity in heterogeneous environments because the resources they contain are not distributed evenly across space. For example, ecotones, or the edge communities that separate two different plant community types, are often more diverse than either of the communities they divide. And theory suggests that these observed differences in diversity are likely because ecotones are characterized by a wider variety of abiotic conditions that originate from the overlap of these two adjacent communities, meaning that they also contain more microhabitats or a niche space for different species to occupy. And it's often obvious how above ground differences in habitat structure can alter resource distributions and availability, such as even in this image on the left here. But it's also important to recognize that heterogeneity in below ground resources can also influence population success, and therefore the composition and diversity of plant communities. So we typically tend to talk about how soil systems vary across regions or landscapes, but it might also be worth considering how soil heterogeneity on a micro scale can influence local species distributions and abundances. For example, previous research from the Henry Lab explored the impacts of soil heterogeneity on the diversity of restored prairie communities. And the results suggest that the edge separating two adjacent yet distinct soil patches, or the microedge as it was termed, may function as its own unique soil microsite, adding to the available niche space in a plant community. And there might even be anecdotal evidence to support this claim. Specifically, a rare tall grass prairie legume, Desmodium canadense, has been seen to disproportionately cluster along the microedge that divides a sandy loam and a clay loam soil patch. So based on this observation of this one species disproportionate clustering along the microedge, we sought to answer the following questions in this project. First, does the microedge function as an additional soil microsite and therefore another separate niche space in patchy soil systems? And second being, what mechanisms allow for a species to establish along the microedge? So there are a few possible mechanisms that could explain a focal species' abnormally high abundance along the microedge, and I'm just going to walk you through these three quickly now. So first is the Goldilocks effect. So if our individual highlighted in yellow here is our focal species, the microedge could feature intermediate environmental conditions that favor the growth and competitive ability of this species, if it does not fare well under the conditions that are present at opposing ends of a soil gradient and also therefore present in the adjacent soil patches. And this is the pattern that we would expect the Goldilocks effect to produce, with individual fitness being maximized along the microedge compared to either of the homogenized soil environments that the microedge intersects. Secondly, when the microedge separates two soil patches of opposing quality and a species is a poor competitor for light, it could be relegated to the microedge where competition for light is less intense, and it can still access essential resources in the high quality soil patch that's adjacent through lateral root growth. In this case, we would expect individual fitness to increase as the quality of the soil environment increases, but in the presence of competitors, fitness would be maximized along the microedge compared to either of the homogenized soil environments. And finally, it's also possible that the observation of a species being disproportionately abundant along soil patch edges is merely just due to chance. And in this case, we would predict that on average, there would be no real advantage associated with an individual's fitness response along the microedge soil environment. So to answer these questions, we conducted a split pot experiment that followed a target neighbor design in the greenhouses at Western University. And our focal species was Desmodium canadense, due to its apparent affinity for the microedge that I previously mentioned, while our neighboring or competing species, Andropogondrardiae and Solidago gensea, were chosen because they tend to be dominant across tall grass prairie communities in southern Ontario and the northeastern United States. So we created a factorial experiment that combined three different soil environments, being a high quality clay loam, a low quality sandy loam, and a blend of the two soil environments that's created by the microedge, as well as two competitive environments, being the absence and the presence of competitors, to create the 6 different treatment groups that you see here. We then planted our focal species in the center of each pot, and we planted our neighboring species based on their environmental tolerances. So that Andropogondrarii was always planted on the left or sand-rich side of the pot, and Solidagogensia was always planted on the right or clay-rich side of the pot. The experiment ran for approximately 3 months, after which we collected the final measurements for different proxies of individual fitness and competitive ability. So I'm first going to talk about the results that we found for our first question that asked whether the microedge can function as an independent niche. I just want to quickly orient ourselves because I'm going to be using the same format for all figures throughout this presentation. So soil environment will always be located on the x-axis and is arranged so that it becomes progressively more sand-rich as we move from left to right. And in this case, we use total biomass as a proxy for individual fitness to assess the response of individuals to the different soil environment and competition treatment combinations. So when we look at our focal species response to competitors in the 3 different soil environments, we see that the fitness of this species is actually greatest along the microedge compared to both the sand and clay-rich soil environments. And this may suggest that the Goldilocks effect is then responsible for our focal species affinity for the microedge. However, when we look at the response of this focal species to the soil environment alone or in the absence of competitors, we see that fitness actually increases as the soil environment becomes more clay-rich, meaning that the Goldilocks effect cannot explain this species fitness response to the microedge, because we would have expected to see the same pattern that we saw in the previous slide, with fitness being maximized along the microedge, even with competitors being absent. So instead, notice that when we compare the focal species fitness response with and without competitors, we see that fitness is greater along the microedge when competitors are present compared to the clay-rich environment, even though this species does prefer this clay-rich soil environment when competitors are absent. This pattern indicates that the soil environment and competition both interact to influence the shape and size of this focal species realized niche along the microedge. And again, just to drive home the point that the Goldilocks effect does not explain this species fitness response in this case, the patterns that we see when we look at this species response both with and without competitors indicate that it's relegated to the microedge to avoid the intense competition for resources that's taking place in either of the neighboring or adjacent patches. So this result elevates the importance of soil heterogeneity in plant communities and also reveals a few noteworthy implications. First, it indicates that edges between compositionally different soil patches can add to the available niche space in a community. It also implies that soil heterogeneity is important for enhancing the establishment of competitively inferior species, as patch edges appear to provide refuge from intense competition. And thirdly, it suggests that introducing soil heterogeneity in restored prairie systems may be an important mechanism for preserving a number of rare prairie species and also the general maintenance of prairie diversity. So now I'm going to move on to talk about the results for our second question about the mechanisms that may support an individual's establishment along the microedge, which we answered by comparing differences in this focal species' ability to acquire resources both above and below ground across the 6 different treatment groups. So first, let's address how light access changed among the different treatment groups. And to do this, we looked at a couple of different proxies for competitive ability for light. But just for simplicity's sake, I'm going to only focus on the focal species shoot biomass today. So immediately we notice that there is a similar trend as when we looked at the focal species isolated fitness response to the soil environment in the previous graphs. So as soil environment becomes more clay-rich, the above ground structures of this species become larger and its ability to compete for and access light also increases. And again, when we introduce competitors, we see that the focal species has greater access to light along the microedge compared to the preferred clay-rich soil environment. And again, just to further reinforce this idea, I'm going to draw your attention to the focal species response in the sand-rich environment, where it appears to fare poorly regardless of whether competitors are present or not. Interestingly enough, both neighboring species also demonstrate this similar response and exhibit poor growth in the sand-rich environment. And additionally, they also seem to perform marginally better in the clay-rich soil environment. So when we put all this information together, we know that all the species in this study benefit from the clay-rich soil environment. But there's also intense competition for light as a result, so that eventually our focal species is outcompeted in this case. We also know that all of our species do not grow as well in the sand-rich environment. So even though our focal species has greater access to light in this case, it will also eventually die out due to a lack of essential resources. But when our focal species exist along the microedge that separates a high-quality clay-rich environment from a poor-quality sand-rich environment, its competitors in the sand-rich environment do not grow as large, so that it's only experiencing intense light competition from one side and is then able to access more light from the other, which prevents it from being outcompeted in the long run. In other words, the presence of soil heterogeneity influences the above-ground structure and competitive dynamics in this community. Next, we looked at our focal species root placement to determine whether it was able to selectively access the high-quality clay-rich patch when grown along the microedge. And to do this, we measured a ratio of root mass that was distributed between each half of all experimental pots, which was calculated in such a way so that small numbers mean that the roots were approximately equally distributed between both halves of the pot, and that larger numbers mean that the roots were disproportionately allocated to the clay-rich side, or in the case of homogenized soil environments, to the right side of the pot. So when we look at our focal species response to the soil environment, we can see that it's preferentially allocating the majority of its root structures to the clay-rich side when it's along the microedge. Whereas in the homogenized soil environments, its roots are actually approximately evenly distributed amongst both halves of the pot. We see the same pattern persist when competitors are introduced as well, which indicates that the microedge provides the focal species with a competitive advantage by allowing it to access resources that are more abundant in the clay-rich soil environment. So there are a number of possible reasons why the microedge may be competitively advantageous for our focal species. In this case, it's actually most likely that there's some sort of resource complementarity that exists between the patches because of the fact that our focal species is relegating the majority of its roots into this clay-rich patch, indicating that there's likely a more abundant resource present there. Alternatively, the microedge itself could also possess unique properties. For example, Stover and Henry proposed that water movement between soil patches may be affected by changes in soil composition, such as pore size, so that water actually slows down and temporarily pools along the microedge. So when we put all of the bits and pieces from this talk together, our evidence overall suggests that our focal species is relegated to the microedge, where it's able to access greater light from the sand-rich side versus the clay-rich side of the pot because of the variation in above-ground community structure that's introduced by the heterogeneous soil environment. And it can also simultaneously access limiting resources that are present in the clay-rich soil environment by preferentially allocating its roots to this higher quality patch. So to wrap up, we found that soil microedges can provide access to resources that may be absent or even too difficult to access in homogenized soil environments, and that they contribute to the available niche space in a community, which may help to support the establishment of a rare prairie species. We've also demonstrated how soil heterogeneity can drive variability in above ground competitive dynamics, meaning that microedges may also function to increase community diversity and therefore have the potential to be an important tool in prairie restorations. But most importantly, we can conclude that soil microedges can actually increase the dimensionality of species niches, both changing the size and shape of a species niche and increasing the amount of available niche space that it is able to occupy in general. And with that, I'd just like to acknowledge some amazing people for all of their help throughout the course of this project. My former supervisor and co-author, Dr. Hugh Henry, and all of the former and present members at the Henry Lab for all of their wonderful support and guidance, as well as Drs. Sheila McPhee, Dr. Greg Thorne, and Jennifer Barron for their great feedback on early editions of this work. And with that, I'm now happy to answer any questions that you may have.
Great talk. We already have our first question from Sebastian. It says, very nice results. To what extent do you think these effects would be lost if the soil heterogeneity in the mixed soil type plots was more finely grained? Or are there things beyond the root growth that matters?
More finely grained. Could you elaborate on what you mean by that?
Sebastian, I'll let you do
Yeah.
that.
At least in the pictures, you had clay on one side and sand on the other, right? So
Right.
that's pretty coarse grained.
Mhm.
But imagine, suppose it was a little more mixed, like the extreme would be very, carefully mix the clay and the sand, so they at smaller scales you see a mixture of both, which obviously wouldn't help with root structure because
Right.
roots wouldn't be able to separate one region from the other.
Right. So I think in that case, you're not really having a distinct boundary. And I feel like having a distinct boundary between soil composition really is beneficial in this case. In one way, you could almost say that the heterogeneity on a fine grain scale is so extreme that it's unrecognizable to plants. So I would almost say that you probably wouldn't see as much of a difference, or any difference probably, in that sort of soil environment versus more of a dramatic difference as we demonstrated here.
Right.
Does that answer your question? Okay.
All right, we have another question from Abdel Halloway. He asked, some studies suggest that plants may increase in biomass like height, leaf area, root mass, while not necessarily altering the number of seeds in response to competition. How do you account for that when using biomass as a proxy for fitness?
Right, that's very fair. So I don't think we can necessarily. One of the limitations of this project is that it was very condensed in terms of time. So we did not actually see all of our plants reach maturity, and we couldn't count seeds, for example. I think that'd be really interesting, though, to explore further in this sort of system to see whether edges actually create more, or soil edges create more of an advantage later on in a plant's life history. Specifically, I guess in this case, we targeted early establishment and how it helps plants to establish in the younger phases of growth. But yeah, I think that's a really great point, and it'd be interesting to further look into that in the future.
I have just a super quick general question, Jessica.
Sure.
So the next time I take a walk in a prairie, how are we defining micro edge versus edge? I apologize if I missed that.
Oh yeah, no, it's all no problem. I guess micro edge in this case, we're thinking more about like how soil patches that may exist within, say, a field, versus edge boundaries typically tend to be more defined along regional gradients, or I'm specifically thinking about where like old glacial, you'll see like glacial differences in soil systems. Those tend to be like larger, more pronounced edge boundaries, like that you would see on a landscape scale, for example, whereas the micro scale would be more so with existing within a region or within a landscape. And I guess we haven't really defined exactly the exact measurements of scale, but probably like within meters to, meter scale are probably the most reliable or easily identifiable micro edge. Just because I think that anything smaller would likely produce more homogeneous soil effects, and anything larger, again, you're looking at more of like a biogeographical context.
I think we've got a quick last question from Dan for Jessica. There's a really nice result that suggests a degree of behavioral habitat choice by plants. I did read behavioral right, in ways that mirror more mobile animals' ability to choose microhabitats. Do you know how sensitive plants are to more quantitative gradients like ratio of sand versus clay?
That's a great question. I'm not super sure. I would assume that it would have to do on their evolutionary histories. For example, one of our actually neighboring or competing species in this case is a very dominant grass that produces these really mat-like root structures. And as a result, it can grow in many different environments because it's able to access pockets of resources throughout a wider geographic range, or larger geographic area. So it can actually persist in lots of different environments. And whereas our focal species is not able to do that because it tends to have this rooting behavior where it selectively allocates its roots to specific areas based on the resources that are available. So Yeah, I'm really not sure how sensitive plants are in general to those minute changes, but I would assume it has to do on their evolutionary histories and those sorts of contexts. It's a great question.
Thank you very much, Jessica. So we'll move on to the next talk from Nicole Adams with a talk titled Population Genomics of a Contemporary Range Expansion in Anna's Hummingbirds.
There we go. Okay. Hello, and thank you for also attending my talk. I'm going to talk about some of the work that I've been doing during my postdoc, as was said, on the range expansion in Anna's hummingbirds. So as we are all aware, humans have altered landscapes globally through urbanization, increasing temperatures and aridity, agriculture, pollution, etc. And what we, or I, often talk about is how these alterations have had negative impacts on plant and wildlife, often resulting in population declines and genetic losses. However, we also know that some species thrive in human-dominated landscapes and are even synonymous with urbanization, as from these rats, coyotes, as shown here, to many invertebrates like ants and spiders. In fact, recent studies have shown that some species of wildlife have been able to adapt quickly to these anthropogenic changes. A study by Whitehead et al. In 2017 looked at environmental pollution tolerance of killifish larvae along the east coast of the United States, seen here. They found that populations from polluted sites, these solid curves, had much higher tolerance to pollutants at concentrations much higher than the normal lethal levels, suggesting that these fish were undergoing rapid evolutionary adaptation to industrial pollution in these extremely polluted waterways. Similarly, a study just last year by Campbell-Staton looked at paired populations of Puerto Rican crested anole from urban and forested habitats, and found that the urban lizards had higher heat tolerance than their counterparts in the forested habitats, suggesting that the increased temperatures in the urban heat islands were rapidly selecting for more heat-tolerant anoles. And with these examples in mind, I turn to our case study, which examines how Anna's hummingbird has responded to human-mediated landscapes as well. In 2017, Greg et al. Documented the expansion of Anna's hummingbird based on community science data from Project Feeder Watch. Annas are native to Southern and Central California, but data from 1990 through 2013 show a northern and eastern expansion, as shown in these three plots of the Western United States. They have expanded up through Washington, Oregon to British Columbia, and further into Nevada and Arizona. The same paper showed that Annas were more likely to colonize areas with higher housing density and were more likely to visit feeders in the expanded range compared to the historical range. As seen in these two plots, the range expansion, this range expansion corresponds with an increase in supplemental nectar feeders over time in the expanded range. The paper's best model for expansion showed that increases in minimum winter temperature were also facilitating the species expansion. Here is their plot of mean minimum January temperature over time in the historical and expanded range. So now we get these adorable pictures of Annas in the snow. 2 years later, C.J. Beatty published a follow-up paper looking at the range expansion over the last century using, again, community science data from the Christmas bird count and as well as museum records. Beatty found that the species has been expanding since around the 1940s, as seen in this map, and has expanded northward all the way into southern Alaska and eastward all the way into Texas. He also found that populations in the northern range appear to be growing exponentially, especially in areas where the winter temperatures are similar to those in the native range. Here we have a predicted habitat suitability over time, and obviously you can see that the habitat suitability increases, as well as an increase in the minimum breeding or winter seasonal temperatures. So he argued that the expansion was due to an ecological release driven by human facilitation, like what was previously presented in the previous study. And this would include introduced plants such as eucalyptus trees and, like we said, supplemental feeders. What remains unknown is the genetic makeup of the expanded regions and whether or not they are genetically adapting to these novel environments. So our collaborators collected over 250 blood and tissue samples from across the native and the northern and eastern expanded ranges. And we conducted low coverage whole genome sequencing on the samples and used genotype likelihoods estimated in angst for our analyses. To jump into results, here we have a PCA that shows samples from the extended range in red circles and the native range in blue triangles. And obviously we can see that they are not very distinct, rather they are quite intermixed, and the variance explained by each PC is only a little over 1%. For funsies, we're going to take the same PCA and just color it by state. And again, we see not much distinction, suggesting that the individuals are contributing equally to the expansion, or at least not one population alone is responsible for the genetic makeup in the extended ranges. To look at this in more detail, I conducted an admixture analysis, which assigns the proportion of ancestry to each individual. In these plots, each individual is a bar, and the proportion of the ancestry group is shown on the y-axis and given by a color. Here, the samples are grouped by state: Arizona, California, Nevada, and Washington. And the native California is shown in blue, and the extended range states are in this red-orange. This plot is based on 2 ancestry groups, and as we can see, it's pretty evenly shared among the states. If we look at ancestry groups 2 through 5, we see similar things. I found that the best supported number of groups was 3, indicated with the star. But again, the groups appear to be evenly spread across all the states, which suggests a very panmictic species across the native and extended ranges. I then divided the samples into 6 groups based on the C.J. Beatty's expansion map, with 3 populations in the extended range and 3 populations in the native range. To investigate divergence, I calculated pairwise FST between the extended population and a nearby native population. I did Washington versus Bay Area, and the eastern samples versus Pacific individuals. And here we have FST with the genome position on the x-axis and this divergence on the y. And in both, we do not see any FST peaks that would suggest that there aren't any regions in the genome that are uniquely divergent between the paired populations. I also estimated nucleotide diversity for all 6 populations. here shown as the mean and the standard deviation. The nucleotide diversity is consistently higher in these middle native populations, groups of individuals, and lower in the extended populations. And this is consistent with what we would expect with edge effects in an expanding population. I then took the difference in nucleotide diversity between our highest nucleotide diversity in those specific populations and found the difference between that and the nucleotide diversity, which was the lowest in the eastern populations. And I found 2 sites that were that showed differences in each population. The site in chromosome 4b, which had higher nucleotide diversity in the Pacific population, is potentially involved in brain functioning, and the site in chromosome 8 which was higher in nucleotide diversity in the Eastern population, is associated with a widely expressed transmembrane protein. And we're looking further into what that means, potentially selection at those sites, if they mean anything. Finally, we conducted an analysis for selective sweeps across the genome for each population. We see a couple of large peaks that show up across all populations, specifically a large pink peak on chromosome 5, but none that are really particular to one population, suggesting that there are no clear expansion selective sweeps going on in Anna's. And this pink peak on chromosome 5 appears to be associated with this TNNT3 gene that is involved in fast skeletal muscle, which makes sense for hummingbirds. So main takeaways, our data suggests that Anna's is a highly panmictic species, and that there has been equal genetic contribution to the expansions. There are potentially areas in the genome under selection across the species, but we found few, if any, genomic regions under selection only in the extended ranges. Therefore, if is there a molecular mechanism underlying the range expansion, it remains unknown. However, for the future, we're interested in using our genomic data to look at genomic by environmental associations to gain insight into a potential molecular mechanism, especially with the environmental connections made in the previous studies. And with that, I'd like to thank my postdoc advisor, Rachel Bay, our collaborators in my funding agency, and thank you. Maybe I went too fast. Time for questions?
Yes, so we already got one. It's like a question from Josh. It's like we just put up a feeder in Seattle, Washington, where we saw a cold and hungry looking Annas outside of our apartment. Second, does seasonal migration impact long distance dispersal for Anna Hemingsbird? Is that something, seems like it would explain panmictic results with a range expansion from the edge?
Yeah, great question. So Annas do migrate. The consensus thought is that they don't do long migrations that you would think for like neotropical passerine birds in North America. They do a lot of migrating in elevation in the breeding season. So the phenology of Annas is still a little enigmatic. Could potentially be contributing to the panmixia as well, yes. but it's not 100% clear where they go and what they do at what time. So more phenology information could really clear that up.
I think this relates to another question by Abdel. I mean, also was curious about how far individual hummingbirds disperse in their lifetime. I guess it's not migration, but more about lifetime. With a reoccurring question of could these be all populations all be mixing?
Yeah, so there are. First off, I do not know the home range. Sorry. But they do, like I said, they do migrate, especially in elevation. But there are some populations that do not migrate at all, which you would think you would see in the genetic data, but I have not seen really any separation of any populations.
Any other questions in the queue, Volka?
No, there's one that came to me, Volker. I'll send it to you.
All right. Yeah, this is a reminder. If you have questions, just send them to me, not Mike. He's like, busy doing technical stuff. So one of the questions was, do you think that panmixia is as old as the origin of overwintering in annas, like 1950s in gardens, or more recent? From Rob.
Panmixia?
Yes.
That's an interesting question. I know C.J. Beatty argued in his paper that kind of the origin of this story is that with like eucalyptus introduction into the Western United States around the mid-19th century allowed the native populations to grow in number, and that then fed into an expansion outward just by having a lot of numbers in the native range, also following human supplementation. So I think to get at that question, it would be really interesting to use genetics of museum samples if you could get them. And then you could really maybe track the mixing over time. But that would be super neat to do.
All right, thanks a lot, Nicole. And thanks for letting me know I need to look for Anna's hummingbirds here in the Gulf Coast of Texas. I didn't know I could see them here.
They're everywhere now.
Winter or summer or both? When should I be looking?
I don't think they breed there, so just passing through.
Okay, all right. All right, at 40 past, we'll have a presentation by Carolyn Dressler titled The Microhabitat Preference of Mated Female Fireflies with and Without Light Pollution.
All right. Hi, everyone. Thank you so much for having me. My name is Caroline Dressler, and I'm currently an undergraduate student at Brown University. Today, I'm going to be presenting some research I conducted this past summer into the microhabitat preferences of mated female fireflies with and without light pollution. And this research was conducted through the Lewis Lab at Tufts University, and I was assisted in great part by Avalon Owens, who is a PhD student in the Lewis Lab. So to start with a very macro perspective, This map here shows the global distribution of the firefly beetle. And as you can see, firefly populations have been documented on every continent of the globe with the exception of Antarctica. And this very wide range encompasses over 2,000 species of firefly. However, over the past several decades, both citizen scientists and experts have noted a decline in firefly populations across the globe. And in response to these observations, researchers have sought to identify what the probable causes of this decline might be. This map here is from a study conducted in 2020 that surveyed expert opinion on several perceived threats to firefly populations and what their perceived severity is. And the threats surveyed here were habitat loss, light pollution, pesticide use, and water pollution. And as you can see, in all but one population considered, both habitat loss and light pollution were thought to be leading threats to firefly populations. So if we've identified both habitat destruction and light pollution to be leading threats to firefly populations, it's important that conservation interventions and efforts made on behalf of these populations aim to mitigate not just one, but both of these threats if they hope to be effective. Now, if we're to start considering how to mitigate habitat destruction of firefly habitats, it's important that we first consider what exactly these habitats consist of. So this image was taken from one of the field sites from which I collected experimental individuals this past summer. And this field site is home to a population of Phytinus obscuralis fireflies that I'll talk a bit more about later. But as you can see in this habitat, it actually includes a great diversity of microhabitat types. So on the left here, we have some shrubs. We've got herbaceous plants. You can sort of see the ferns and longer grasses there. We have mud and wet grass in this marshy area. It's a little bit harder to see. We have some tall grasses here. Some dried grass that is below the taller grasses in this whole area. And then it's not visible in this picture, but there is also a section of mowed grass that I was actually standing on when I took this image that is also accessible to fireflies in this population. So this great diversity of microhabitat types is typical of a lot of firefly habitats in the United States. And if we are to consider which microhabitats we want to conserve for populations, it's important that we know which microhabitats these fireflies actually occupy. However, identifying the microhabitats that these fireflies occupy is a little harder said. It's a little easier said than done. And to explain that, I'll talk a little bit about firefly courtship behavior. So if we were to linger in this particular habitat until dusk, this particular firefly species comes out a little bit after dusk. And what you'd see is a lot of males flying about and flashing. Now, if you've ever seen fireflies out in the wild yourself, or maybe you've seen one of those long exposure shots of many of flashing lights against a dark background, or perhaps you've even, as a child, collected fireflies and kept them in a jar in your room for several days. All of those fireflies that you would have seen or interacted with would have been male fireflies. And that's because as part of their courtship behavior, the male fireflies will fly around and flash very visibly in order to communicate and attract the attention of female fireflies that are lower to the ground and much harder to identify. Now, the females may flash once or twice in response to the males, but generally, as a human observer, it's very hard to find them. And because male fireflies are so visible and much easier to observe, conservation research up to this point that has been made on behalf of firefly populations has really expressly focused on the male firefly. There is a lot that is known about the male firefly's courtship behaviors, its flash patterns, its microhabitat preferences, and more. But in contrast, very little is known about the field behavior of female fireflies. However, the field behavior of female fireflies is critically important to the conservation of the beetle as a whole, because it is the microhabitat preferences of female fireflies that will directly determine the microhabitats in which the eggs will hatch into larvae that will ultimately develop into the next generation of adults. And what's even more interesting is that fireflies only spend about 2 to 3 weeks of their full life cycle as adults, whereas they spend 1 to 2 years in their subterranean larval stage. And so embryonic development and the larval stage together account for over 98% of the total life cycle of both sexes of fireflies. And we do not know where this life cycle takes place. We do not know the microhabitats in which eggs and larvae can be found. And if we were to know more about the microhabitat preferences of females, that would greatly help us unearth where over 98% of the firefly life cycle takes place, which would be very informative for conservation efforts. So this brings me to our experiment from this past summer. We designed an experiment to assess the microhabitat preferences of 3 species of fireflies in the Phatinus genus. And the Phatinus is a leading North American firefly genus. And the 3 species of Phatinus that we investigated were Phatinus obscuralis, Phatinus aquilonius, and Phatinus marginalis. And we also sought to observe how and whether the presence or absence of light pollution affected their microhabitat preferences. So our experiment proceeded in 3 phases, corresponding to the 3 species of Phatinus firefly that we studied. The first phase focused on the species Phatinus obscuralis, and they were collected from the field site shown here. And also from this field site, we collected 5 microhabitat choice options. And these choices included mud, moss, about 2 to 3 centimeter grass, dried grass, and herbaceous plants. Similarly, for our second species, Phatinus aquilonius, collected from this field site here, we also collected 5 microhabitat choices, including mud, moss, about 2 to 3 centimeter grass, dried grass, and herbaceous plants. And finally, for Phatinus marginalis, the third species, collected from this field site shown here, we also had 5 microhabitat choices, which were, again, mud, moss, grass, dried leaves, and herbaceous plants. Once we'd collected these microhabitat options, we then inserted them into our experimental arena in the lab. The arena was about 110 centimeters in diameter and was surrounded by a plastic wall that was about 30 centimeters in height. The arena was subdivided into 10 wedges, and so each habitat choice was represented twice, once with a light treatment and once without. And to control for any differences between the 2 versions of each microhabitat type, we rotated the light by 1 habitat wedge every night so that over the duration of the entire experiment, each microhabitat version was both with and without the light treatment. At the center of our experimental arena, we had a choice platform from which we released a new cohort of individuals every night. These individuals were marked individually using fluorescent powder so that in subsequent days during our observations, we could locate them using a UV light. Now, after releasing individuals, we observed them 3 times daily for 30 minutes. And during these observations, we would chart their location, so which microhabitat they were in, how far into that habitat they were, and how high up they were. And so here on the left, you can see our experimental arena. This, I believe, was for the 3rd phase of our experiment, so for the marginalis fireflies. And you can see the 5 different habitat choices, each represented twice. So in this phase, we had herbaceous plants, moss, grass, mud, and leaves. And on the right, you can see an individually marked firefly that has been relocated in the arena using a UV light. So now I'm going to go over the results that our study uncovered. All right, this plot is oriented as if you are looking down vertically into the arena. So here you can see each microhabitat represented twice, and each point that will appear on this graph corresponds to 1 unique position in which a firefly was found during 1 observation period. And again, the fireflies were observed 3 times a day over the course of the experiment. The triangular points correspond to females that were observed in the control-treated microhabitats, whereas square points correspond to females that were found in the light-treated habitats. And I'm going to go female by female. So this one, you can see she tried out grass and herbaceous plants. She was most frequently found in the control-treated plots, and she seems to exhibit a preference for grass. This female exhibits a similar preference. This one seems to have sampled a few more habitats, and we can continue layering on the different females in this way. Oops. And what we see at the end is that overall the females did sample different habitats, but they seem to prefer herbaceous plants and grass over, for instance, mud and moss. And these results were for the Phytinus aquilonius, which were the second species that we studied. And if we make a similar graph for all three species, we can kind of see that across the three species, these trends hold true, where grass and herbaceous plants appear more popular than do moss and mud. Now, for the remainder of this presentation, I'll focus my analysis on the second species that we studied, Phytinus aquilonius. So we analyzed our data using a binomially distributed generalized linear model, and this graph here will show the results that it generated. So on the y-axis, we have mean choice, which is just the probability of finding a female in a given microhabitat. And on the x-axis, we have the 5 microhabitat choices. So for the control treatment, we found that herbaceous plants in grass were significantly more likely to contain a female than were dried grass, moss, and mud. In other words, females significantly preferred the herbaceous plants and grass over dried grass, moss, and mud. And if we add the results for the light-treated microhabitats, we find that the same trends hold true with the exception of herbaceous plants, where we were much more likely to find a female in the control-treated herbaceous plants than in the herbaceous plants under the light treatment. So to summarize our results, we found microhabitat to be a significant predictor of where a female was likely to appear in our experimental arena, in contrast to light treatment, which did not significantly predict the location of a female in the arena, with the exception of the herbaceous plants, where again, we found that we were much more likely to find a female in the control-treated herbaceous plants than in the light-treated herbaceous plants. In the control treatment, clipped grass and herbaceous plants were significantly more popular among the females than were any of the other microhabitat choices, and in the light treatment, clipped grass alone was significantly more popular than any of the other microhabitat choices. So one possible explanation for these results is that in the evening, the preference for herbaceous plants could be due to the fact that female fireflies tend to want to crawl higher up in order to attract the attention of a potential mate. And the fact that they might want to have a more contrasting light environment could be that they want to make sure that their response flash to the flying males that will be flying above them will be more visible. Once again, our research has demonstrated that female fireflies do in fact exhibit microhabitat preferences. And future research in this area might investigate more specifically where female fireflies, what microhabitats female fireflies prefer to lay their eggs in, because these microhabitats will even more specifically indicate where the larvae are likely to develop and where that other 98% of the life cycle of both sexes takes place. And once again, research into the microhabitat preferences of female fireflies is critical to the conservation of the beetle as a whole, because where these females go after they mate does determine where the larvae develop and where the next generation of adults will appear. So thank you so much. I'd really like to thank Dr. Lewis of the Lewis Lab, Avalon Owens, who helped me so much, Tufts University, and everyone at ASN for making this conference possible. And I'm happy to take questions if there are any.
Yes, there are questions. We got a question from Avalon. And so he's asking, did the females seem to react to the UV light that you used to find them?
I didn't notice any reaction to the UV light. Typically, when I would find them using the UV light, they wouldn't move. Female fireflies tend to move quite a bit less than do the males, especially during the morning and evening observation periods. But that could be something to try to control for in future iterations of this experiment.
I have a quick question, Carolyn. Do you think so you were measuring like their presence where they were, you weren't measuring whether they were laying eggs in any of these habitats, is that correct?
No, it was hard to tell. I could see that they were laying eggs, for instance, when I found them in the mud, but it was really hard to tell when they were in like the herbaceous plants. The eggs are really hard to locate with the UV light. Yeah.
Right. So do you think they were, preferring these areas for thinking about oviposition and where their offspring would be living? I mean, are there differences in the food that the larvae eat between these microhabitats?
Right. Yeah. So originally this was inspired by the fact that last year I'd found a bunch of Phytinus larvae in sort of a muddy bank type area. So I initially expected them to prefer the mud because it seemed to be where I'd found so many larvae, but in fact they didn't. So I don't know, it could be because I was not observing them laying eggs, like maybe they really were still looking for mates at the time that I put them into the experiment. But yeah, I agree. It would be really interesting to sort of redesign the experiment to specifically look at oviposition.
We have another question from Katja, who's asking, do you have any idea whether microhabitat preference in the lab roughly corresponds to microhabitat use in the field?
Yeah, so that was also something we'd considered, and something I might do if I do a follow-up version of this experiment is if we could have sort of a side-by-side, like where do we find them in the field, what microhabitat are they on in the field, and then where do they go in the arena. For the last portion of it with the marginalis, we did try and notate what kind of plants we found in the field, but I haven't actually tried to see how that matches up to this data yet. But yeah, I agree. That's a really interesting comparison to make.
All right, I have one more natural history question.
Mm-hmm.
What do your specific species as larvae eat? I mean, do they specialize on worms, other invertebrates in the soil? What are they consuming? Do you know?
As far as I know, they consume worms. When we reared larvae in the lab of Phytinus obscuralis, which is one of the species that we studied here, and Phytinus, the genus in general, tend to thrive on worms, as we found.
Thanks. All right, we have like 1 or 2 minutes till the next talk. Thanks, Carolyn.
Thank you.
All right, at the top of the hour, we have Nick Crouch, who will be giving a talk titled The Role of Geographical Co-occurrence in Driving Lineage Diversification in Terrestrial Vertebrates.
Okay, hi everyone. Thank you for coming to my talk today. First of all, I want to address why the title on the slide is slightly different to the one that Scott just introduced. It's just to match up with the title that's actually on the manuscript that's in revision to hopefully sort of give people more of intuitive way of finding it when it comes out. So today, I'm basically be talking about how we can use the present day geographic distributions of species to make inferences about the evolutionary processes that lead to the formation of species, as well as try and understand what aspects of biology may limit diversity within certain regions. So this topic comes under this broad umbrella of why do we see the species that we see around us today? And this is, a very sort of classic and ongoing question that troubles a lot of people, is that across the tree of life, we have an excess of both species rich and species poor clades. And actually species poor clades are more overrepresented in the tree of life than these are very speciose clades that, it's very intriguing to try and find out what's driving that diversification. But in order to try and get this sort of balance of understanding mechanisms of diversification, I think it's important to sample across the tree of life and sort of capture the variation we see across these different clades. And when we think about speciation events, when we think about diversification in general, that always has a spatial component to it, the geographic world in which these processes occurs. And so within the different ways in which new species form, there's a lot of evidence, particularly in birds, which is sort of my predominant field, that allopatric speciation is much more common than sympatric speciation or other forms in which speciation can occur. And that's because there's a very physical interruption of gene flow between distinct populations, and that given that these populations are now occurring in different habitats, local adaptation to those different conditions may also facilitate these populations to diverge, such that if they come back into secondary contact, enough differences have accumulated that they can be sort of defined as good species. But we know that diversification cannot be unbounded. So first of all, the Earth is a bounded space, it's finite. And that way, in this regard, lineages may simply run out of room with which to create new species. This is going to be particularly problematic for species that have exceptionally large ranges. So even though it's a sort of weird species complex, these avian species I've highlighted are particularly big ranges. There can also be ecological limits to diversity. And in this, what I think is a classic paper by Roboski and Levett, they hypothesized that as new species continue to form and continue to utilize the available resources that are available within an area, diversification will begin to slow as more and more of those resources are used up, and there's less opportunity for new species to form. So within the framework of existing theory that we have, it allows us to sort of make some general predictions about how we think different factors will impact lineage diversification. And so because we think that allopatric speciation is both more common and because it will have this immediate cessation of gene flow, one hypothesis that you can make is that if you have a greater proportion of your lineages within a taxonomic group in allopatry, you would have more species overall within that clade because there's more opportunities for those species to form. But as we sort of just thought about, there are ways in which you can deviate from this prediction. So first of all is that limits to diversity that, say, competition with other congeneric or confamilial species may place on that relationships. There might not be the ecological opportunity within the environment for which that new species can form. Alternatively, you can get bursts of lineage diversification that are completely unrelated to the geographic distributions of species. And again, there's been a lot of really interesting review papers coming out in the last few years that really sort of document there can be a myriad of processes by which new species can form in this way. And in this work, I just highlight the work by Mendelsohn and Shaw from 2005 that showed that this genus of crickets, to give some love to invertebrates, really had accelerated diversification due to selection for secondary sexual characteristics. So this really sort of forms a lot of the conceptual framework that I sort of tried to tackle this work, but it's important to note that this is not an exhaustive list of variables that can affect diversification. These were chosen because there's basically, we can get data on them, which is an important consideration for macroevolutionary studies, but also because we have this rich history of previous work that allows us to evaluate new results in the context of historical work. And so there are a lot of interesting variables, such as, is the effective population size of species different in allopatry and sympatry? That's just really difficult to get at without a sort of a very dedicated study. So allopatry in itself is a very easy to understand term, yet there's different ways in which we can define it. And so in this work, I use two approaches. So first of all, I should say that the main taxonomic level of this work is the family, but I have species level and family level definitions. So first of all, we're going to start at the species level, and we're going to define allopatry on the left-hand side first, only using the relationship between a species and its sister taxon. So this is following basically Weir and Price, who assigned geographic states to nodes within a phylogeny based on the descendant taxa. Another way of thinking about that is you can define allopatry for any given species based on its geographic relationship to any other species within its family. So if we look at species C in its sort of rectangular box, which defines its range, on the left-hand side, it's defined as allopatric, but on the right-hand side, it's sympatric because its range overlaps with species D, even though that's not its sister taxon. Phylogenetic data for this work comes from three very large studies that have been published fairly recently and slightly longer ago in the case of the Yates et al. Study. So an important point, which I'll come back to later, is that taxonomic data is used in the construction of these phylogenetic data. That's simply because of the challenge of creating phylogenetic data covering the taxonomic diversity of these groups. In the case of the bird and the amphibian data, I reran the placement of those species within a genetic backbone using the TACT method of Chang et al., which came out last year, which calculates speciation extinction rates for each designated clade within a tree to try and get some more realistic branch length estimation. And then for all three, I ran independent BAM analyses to get lineage-specific speciation rates. And then I used STRAP to compare the speciation rates for only extant taxa, so we don't have to deal with this problem of unknown extinction rates, using STRAP, which is a very robust way of testing for correlations between a rate and a trait of interest. Here, because I've got a binary trait, I specified the Man-Whitney-U metric, and all of the rates took the difference between the observed and a null generated by permuting the rate values at each of the tips. So that's with the species level. Moving out to the family level, I had two definitions of allopatry or geographical co-occurrence at this level. So first of all, what is the maximum single diversity that you have within a geographic area? So this is sort of following Weir's analysis of neotropical birds from 2006. But this definition kind of doesn't capture all of the variation that we see. So I also followed a recent paper by Kennedy et al., which takes the mean value of all those overlapping ranges. So it sort of is a more sort of overall view of family co-occurrence. And I compared these against two metrics of diversification histories. So first of all is this very commonly used gamma statistic of Pybus and Harvey, which calculates the relative position of nodes within a phylogeny compared to that under a pure birth process. So here those nodes are kind of shifted towards the root, indicating a potential slowdown in diversification in this clade. This metric has some problems in that it doesn't scale entirely comparably between phylogenies of different sizes. So I also used this statistic from Piggott et al. From 2010, where you compare the diversification history in the first half to the second half of the clade's history. where that's just the change in the number of lineages over time. And here it really sort of emphasizes that slowdown since no new lineages originated in the second half. Broadly, these two metrics were sort of broadly correlated with an R of around 0.6. So looking at the effect then of our species level definitions of allopatry on speciation rates, when we define allopatry using sister taxa, we find consistently across these three clades a higher speciation rate in allopatry, which is kind of a nice proof of concept in a way, because it would have been a bit concerning if that wasn't true. So if we compare and contrast that now, when we use our second definition at the family level, we find contrasting results. For mammals and amphibians, we now find that they have a slower rate of speciation in allopatry, but birds maintain this apparent higher speciation rate in allopatry. But there's an important difference between the avian taxa here and the mammals and amphibians, is that the vast majority of these avian species, which are still classified as allopatric under this definition, are almost all of them, or 3/4 of them, are obligately island-dwelling species. And so this can impact our interpretation, because is it now dispersal to new islands and the introduction of geographic barriers that leads to species formation? Or, as some recent work has shown, is it changing sea levels, severing connections between populations that causes diversification? So sort of raising some additional questions. If we look at our family level results now, so each point, there's a lot of dots in these panels that I'm going to show on this slide and the next slide. Each point is an individual family, but the main thing to look at is the general trend lines. So first of all, showing this expectation that more families in allopatry, which is on the x-axis, should correlate with more diversity overall. Well, if we look at the definition using sister taxa, we just find no relationship at all. It's just a buckshot of points. However, in the complete opposite to what was predicted, we find when we define allopatry using all confamilial taxa, if you have more of those species in allopatry, you actually have less species within that family overall, which is very, very interesting. I haven't shown it here just for simplicity of the slide, but I also tested for the deviation through the speciation rate or slowdowns. There was no correlations either with the residuals from regressions or absolute values. So speciation rate and diversification slowdowns don't really seem to be important at the family level. I also want to note that on the right-hand panel plot showing this negative relationship, for families that had no allopatric species, they spanned the full range of diversities that we see in family species richness. So I kind of think that that suggests that there are external factors, such as perhaps evidence showing that weird morphologies in birds may also regulate diversity. So it kind of, because the results are counterintuitive, it made me want to sort of have some sanity check aspect to this work. And first of all is that we expect that more diverse families should overlap more with each other. They should have more overlapping ranges. And that is what we find in this top row. So more overlapping ranges correlates with higher species richness. But in the bottom two rows, where the rows correspond to gamma and p statistic, and the left and right columns again correspond to our two definitions of allopatry, there's no relationship between slowdowns and clade diversity. So because this contrasts with previous work, I kind of feel that this shows that slowdowns are idiosyncratic properties of certain lineages rather than broadly applicable trends. I also want to take the time to highlight two principal concerns. First is that the taxonomic data can be extremely problematic with trait analyses on the resulting phylogenies. However, I want to acknowledge that each of these three clades will have their own taxonomic practices, their own sampling biases, their own idiosyncrasies, yet we're still able to detect a common pattern across the three clades. So that to me suggests that even though there is undoubtedly noise, we can still identify some signal. Second is that, and we've seen some talks on this earlier today, range sizes are extremely flexible through time. The house sparrow example was particularly striking. And so on this graph of changing temperature through time, I've highlighted the mean ages of nodes that subtend 2 extant species from each of the 3 major clades. And as you can see, they significantly exceed the particularly dramatic climatic fluctuations and therefore presumably range fluctuations that happened during the Pleistocene. And there's also evidence from the geologic record that range sizes can be labile. That's harder to sort of take into account, I think, for analyses like this. So to bring all of this together, what do I think is going on? So first of all is this sort of nice reassurance that geographical separation of sister species does facilitate more rapid speciation. But I think that what these results show is that in order to achieve maximum diversity for any given family, it seems to be the ability to tolerate each other or pack within the same geographic area that allows diversity to accrue. And I'm really excited to keep exploring this question using data on functional groups and functional traits to sort of more explicitly test this hypothesis. And then finally, I think that slowdowns in diversification are sort of idiosyncratic properties of certain lineages, potentially affected by morphology, where there's evidence in birds again, but that's the case. So I would like to thank, first of all, the organizers for all the work, effort, and dedication that's gone on to making this whole event happen. I'd like to thank Drs. Price, Jablonski, and Eddy for their always fun, informative, and productive discussions. The Jablonski and Price labs also gave some feedback on this work, and I'll happily take any questions that anybody has.
Great talk, Nick. Nick, I'll jump in with a real quick question. You just addressed it, which is just the distributions of your species now are not the distributions of your species in the past. Could you expand a little bit more about how you're going to deal with that, whether you need to deal with it, or how that issue can be addressed?
That would be the part of it where I'm least sure that how to address it, whilst being most confident that it needs to be addressed. Mm-hmm. So, one possibility is doing habitat or distribution reconstructions going back through time. The problem being is that our confidence with those is just going to drop off through time. And ideally, we would need those over 2-3 million years. So, yeah, very important, but I am open to suggestions from our community as to how best to potentially address this. Maybe it's deal with extinct, use only the fossil record, but then we have to deal with taphonomic biases and things like that. So open to suggestions for sure.
Mm-hmm.
We have a question from Inbar, and she's asking whether you're considering the present day distributions or historical distributions like ancestral range reconstructions for your species and family analysis. And how do you think those two distributions are related, and how would they impact the geography of speciation?
So it's definitely just the modern day distribution. So these data probably reflect distributions within the last 50 years. I think we could probably get good estimations deeper in time for a smaller proportion of the species, but I don't think enough for a broad macroevolutionary analysis like this. So I think it would be a sort of clade specific answer. I think I'm kind of getting short on time, so if you want to shoot me an email or something, I'd be happy to sort of talk about that further.
Yeah, thank you very much, Nick. Our next presentation is a pre-recorded presentation from Paulo Guimaraes, titled The Structure of Ecological Networks Across Levels of Organization.
Good night. First of all, I would like to thank the organizers for putting together this virtual meeting, and thank you for watching this talk. It's a pleasure and an honor to be here. Today, I would like to talk about the structure of ecological networks across levels of organization. And to do so, I would like to invite you to visit an ecosystem nearby Asilomar, the kelp forest. This species-rich system may illustrate the role of ecological networks at different levels of organization in contributing for biodiversity. In similar abiotic conditions, this system can shift to a species poor system dominated by sea urchins. And this shift is related to the absence of an ecological interaction. If sea otters are present and prey upon sea urchin, then kelp forests are maintained by an indirect effect. An indirect effect occurs through a chain of direct interactions. In this particular case, sea otters reduce the numbers of sea urchins, and as a consequence, kelp forests increase in biomass and sustain a species-rich system. This sort of impressive indirect effect can only occur in a network. Networks are pathways that allow these indirect effects to propagate. And a central question is to understand how these interconnected networks emerge in ecological systems. We know a lot about the kelp forest system due to the work of multiple research groups, especially the work by my good friend Jim Estes. A couple of years ago, Jim drew this ecological network describing the main interactions of kelp forest. Is now framed in my office in São Paulo, and the red square identify the three species I mentioned before: sea otters, sea urchins, and kelps. And sea otters might be central to this system because they interact with multiple species. They are highly connected species, and because of that, for example, they are not they not suffer with negative feedbacks of reducing the population of one of their prey. That some kind of dynamics that we would expect in like if the species is a specialist in a single resource. But if you zoom in a population of sea otters, you realize that not all individuals are identical, but they feed on different resources. They vary in the degree of specialization. There is, in other words, intrapopulation niche variation. This intrapopulation niche variation can be described as networks, individual-based networks in which each individual is, in this case, a square. These circles represent resources. These individual-based networks may provide insights in different ecological and evolutionary dynamics of populations. But here, I'll focus in the role of individual-based networks in generate, at the community level, interconnected networks. To understand how individual-based networks may contribute to the emergence of connected networks at the community level, and consequently of the presence of indirect effects. First, let's consider here some examples of theoretical examples of individual-based networks. The squares are individuals, the circles are resources like prey or mutualistic partners. These different individual-based networks lead at the species level to different species subnetworks. Please note that there is only one way of generating a species subnetwork, as in E, but there are multiple ways of generating a species subnetwork in which one species is interacting with multiple partners. These species subnetworks combine to each other, forming species-based networks. And there are two main forms of networks, not only in ecology, but in different systems. Disconnected networks and connected networks. Disconnected networks are formed by multiple components, groups of isolated species, and a connected network is characterized by the presence of what we call a giant component, a group of species that are interconnected through direct or indirect pathways. These two different forms of networks are observed in ecological systems. Here we have examples of disconnected networks as interactions among plants and galling insects, and among ants and myrmecophytes, and connected networks as interactions among prey and predators, and among plants and frugivores. These two forms of networks, they represent different phases. And this is not a metaphor. This is actually, theory predicts that these two different networks, disconnected and connected networks, are actually two different phases separated by a phase transition, as ice and water, as iron and magneto. To understand this phase transition, let's consider this plot. In the x-axis, we have a connective parameter. I will not go into the details of this parameter. I'll be happy to do that after the talk. But it's essentially, as you move from negative to positive values, you move to low variance in the distribution of interactions per species to high variance in the distribution of interactions per species. In the y-axis, you have the proportion of species that is part of the main component of the network. So as you go close to 1, all species are linked to each other by direct or indirect interactions. 0 is the predicted critical point derived by a theory of random graphs, random networks. So values higher than 0 will lead to a connected network, values lower than 0, a disconnected network, and we should expect a sharp transition around 0. And indeed, empirical species-based networks are follow these predictions, and no matter if they are mutualisms or antagonisms, they obey this architectural law that is predicted by random graph theory, in which each circle here is an empirical network. The red circles are networks predicted to be connected, supercritical networks. The black circles are predicted to be disconnected. And as you see, you have a very sharp transition between the two phases. And as the value gets more and more positive for the connective parameter, the network becomes more and more interconnected. This is a result of increasing variance. It is increasing variance in the number of interactions per species is a result of the presence of a few highly connected species. So a fundamental question is, what makes a species highly connected? We know, for example, that species abundance vary very widely across the species in an ecological community. And indeed, there is a correlation between abundance and the number of interactions in a given community. But we also have many examples of species that are not particularly abundant, but are also highly connected. Many of the keystone species are highly connected species and are rare species. In this case, traits may play a fundamental role in explaining why some species are highly connected. And indeed, if we analyze the traits and how traits filter out interactions, we can get an understanding about why some species, some close related species, vary widely in their number of interactions, as here illustrated by two Sylvia birds in Spain. There's 2 species from the same genus. They vary in their number of interactions, basically because Sylvia undata is constrained by the size of fruits they are feeding, whereas Sylvia atricapilla is able to feed in most of the fruits available in the community. If traits are mediating the emergence of these highly connected species, we can try to connect the evolution of these species interactions to the emergence of connected or disconnected networks. For instance, disconnected networks are only predicted to emerge if selection is favoring extreme specialization, or selection is against generalization in all species in an interacting assemblage of plants and herbivores or mutualistic partners. In contrast, if selection is favoring some species to be highly connected, then a connected network will emerge. And these highly connected lifestyles are species that are actually very specialized in the network, not in particular species. Those are hypercarnivores that feed on dozens of different prey species, mutualistic partners that interact with multiple plant species across the communities, as honeybees, as some frugivorous birds, as some toucans, some Camponotus ants that visit extrafloral bearing plants, cleaner globies interacting with dozens of clients in coral reefs, and obligated scavengers that feed on carcasses of different species in a given community. This is very specialized lifestyles. They create the network that allow indirect effects to propagate across the system, creating huge cascading effects. So we saw that individual-based networks that fuel the emergence of highly connected species may, as a side effect, generate also a connected network at the community level. And this may be one example of how this hierarchical view of ecological interactions, in which we start to realize that generalism at one level of organization emerge as a consequence of a collection of specialist and generalist units at a lower level of organization, may provide insights about the structure of biodiversity. And this idea may have consequences to larger spatial temporal scales. This is a picture that was took by a friend of mine nearby my lab at São Paulo. This is my favorite tanager, Traupis ornata, feeding on fruits of a Chinese plant. And due to human activity, we are now observing a big interchange of species across the globe. And now we are generating highly connected species at the scale of the entire globe. And this is beautifully illustrated by this amazing work by Frickel and Svenning, in which here we have a global network of plants and frugivores. And the interactions of these plants and frugivores, if you focus only on the native species, the network A, you can see a coarse description of the geographic structure of the network. But if you consider also the interactions among introduced species, invasive species, species that were benefited by humans and move them around, these spatial structures start to be erased. This is the effect of highly connected species on the structure of this network at the scale of the entire planet. We do not know the consequences, if any, of this change in structure, but we could predict, or we could expect, that this change, this increasing interconnectedness will favor cascading effects across larger and larger spatial and maybe temporal scales. I would like to finish this talk thanking all the collaborators, the members of my lab, my department and Universidade de São Paulo in Brazil, and all the funding agencies that support our work. If you want to know a bit more about our work on network structure in ecological systems across levels of organization, this review is now out since last November, and I am glad to answer any questions. Thank you so much for your attention.
Thanks a lot, Paolo. And I believe while that was pre-recorded, Paolo is here to answer questions.
Questions are coming in. We just need to wait a little bit. So I guess I can see them typing. Hey, we've got a question from Tad. He's saying, great talk. I was wondering about the different meanings of interactions at individual and species level, and if this gets in the way of trying to incorporate things like interaction strength or gain understanding.
Okay, thank you for the question. I think that this is one of the main challenges if we want to try to integrate how the resource used by individuals within populations translate to or affect the population dynamics of different populations interacting at the community level. So I really do not have an answer for that. We know that averages, that the way we usually use when we describe interaction strengths at community level, may oversee the consequences of variance within populations to the ecological and potentially the evolutionary dynamics. So as always, when we talk about networks, our first problem is describe the structure, and the second problem is how to connect the structure with dynamics. And this is a problem of change, the currency when we move across the scales.
We got another question by Brian. He's asking, is there any reason to believe that disturbances could drive phase transitions in the networks?
Yes, there is, but of course the evidence now is only anecdotal. For example, we know that some ant-plant interactions that usually form groups of isolated pairwise interactions or small groups of interacting species in the Amazon forest. When you go to the forest edge, some generalist ant species take over the system and they connect groups of species that would otherwise be isolated. So in a physical point of view, we're just moving the system from a disconnected system to a connected system. And then one possible consequences are the indirect effects now start to cascade through the different otherwise isolated groups.
Thank you very much, Paolo. If you have any more questions for Paolo, you could put those in the chat or the Slack channel. Our next and last speaker of the session is Matthew Comelford, and his talk is titled Spatial Sorting Drives Rapid Adaptation and Maladaptation in the red-shouldered soapberry bug?
Oh, sorry, couldn't unmute. Thanks, Scott. Hi, everyone. Thanks for hanging in there to see my talk. It's been a long but exciting day. My name is Matthew Comerford, and I'm in my last year as a PhD student in Scott Egan's lab at Rice University. Today, I'll be sharing with you a project that me and Scott have been working on in collaboration with Scott Carroll, and that is a large-scale natural experiment that places dispersal and spatial sorting in an evolutionary framework by testing its capacity to drive directional shifts in traits that go beyond simply dispersal ability. So traditional Darwinian evolutionary thinking predicts that adaptive traits evolve due to selection because they facilitate organismal survival and/or reproduction from a set of genetically based phenotypes. And thus we think of evolution as a phenomenon that occurs across generational time spans. In similar fashion to selection, spatial sorting has the potential to create a shift in some phenotypes within the population, as it is the process in which variation in dispersal ability is sorted across density clines. However, this is not a function of fitness, but rather because better dispersers have a higher probability of sexual encounters with strong dispersers as they move away from the origin of the population's home range. Thus, spatial sorting has the capacity to produce instantaneous changes in allele frequency at the population level, making it a unique evolutionary mechanism as it occurs across space rather than time. Spatial sorting and differential trait dispersion have been a common theme in invasion biology, but as of recent, they've garnered greater interest within the world of evolutionary biology. Biologists first started associating spatial sorting as an evolutionary process as a result of a series of publications by Richard Shine and his colleagues' observation of the cane toad invasions of Australia, such as this figure we borrowed from Rollins, Richardson, and Shine's 2015 paper in Molecular Ecology that depicts the westward invasion of the introduced toads along the East Coast, which exhibit a 5-fold increase in its dispersal capacity as their invasion front moved across the northern region of the continent towards the aptly named town of Darwin. Natural observations of spatial sorting's evolutionary potential have been since corroborated by lab-based experiments and modeling, notably by Brad Ochoki and Tom Miller using bean beetles and their dispersal into a matrix of artificial patches. So here on the x-axis, we have the number of patches a cohort of beetles disperse into, and on the y-axis represents the density of beetles within each of those patches. Each color represents a subsequent generation. The spatially sorted cohorts were allowed to mate with other individuals who dispersed into the same patch, while the homogenized groups were randomly mated. And what we see is that within 10 generations, there's approximately a 9% increase in dispersal ability among the strongest dispersers in the spatially sorted populations. So how does this happen if dispersal is not correlated with fitness? The process of spatial sorting occurs when there is a phenotypic variation in traits that affect dispersal ability. For instance, in this theoretical home range, the green square, we have a population that expresses 2 discrete phenotypes, the blue circles, which represent different dispersal abilities, darker circles being the better dispersers. Traits that enhance the rate of dispersal accumulate at the leading edge of the expanding range, or the newly colonized habitats, leaving weaker dispersals behind to populate the range's core habitat. As mate choice becomes limited in the sparsely occupied new range front, a sortative mating occurs between these high dispersing individuals, which then drives an additional increase in dispersal phenotypes in subsequent generations, leading to dispersal phenotypes to become spatially sorted at the range's edge, which we saw in the cane toad example. With spatial sorting being well tested in its ability to manufacture increased dispersal rates as a result of differential mating by dispersal ability, I wanted to advance spatial sorting one step further in our understanding of its role in evolutionary theory. I will do this by testing how spatial sorting might facilitate or subdue rapid ecological adaptation when traits that aid in dispersal happen to be linked to important fitness-related traits in an ecologically divergent landscape. To do this, we'll take advantage of the red-shouldered soapberry bug, a dispersal polymorphism, and an extreme episodic climate event. Due to the original work by our collaborator, Scott Carroll, populations of the red-shouldered soapberry bug in Florida have become a textbook example of rapid ecological adaptation in response to host shifts in an introduced plant with narrower seed pods in which they feed. Consequently, these insects evolved shorter beak lengths in less than 40 years. As part of my dissertation, I've continued exploring this rapid ecological adaptation story in a region of the country in which we see a lot more complexity, as there is a greater number of host associations under varying degrees of geographic overlap, which represent ecologically divergent landscapes in our hypothesis. So, what background information do you need to know in order to convince you that this system is an effective means of testing this hypothesis? First, you should know that in Houston, all three of the soapberry bug's primary hosts occur in sympatry, and that the rain tree, balloon vine, and soapberry all have different seed pod depths. So here on the inverted x-axis, we can see that each of these three host plants, and on the y-axis, we have the distance the insect must penetrate with the beak-like proboscis to access the seed for consumption. And as you can see, we have this pattern where balloon vine has the greatest depths, while rain tree has the least amount of depth in which the insect must penetrate. To be successful, insects require beaks that match their given host association. So here, again, instead of on the y-axis, we have the mean beak length of the insect itself. And then on the x-axis, we have the three different host associations. And what you can see is this pattern is matches the pattern we saw before, where the longest beaks belong to balloon vine associated insects, and the shortest beaks are on the rain tree associated insects. And thus, seed pods are the selective pressure driving rapid ecological adaptation of beak length for the soapberry bugs in Houston. Second, soapberry bugs has a discrete dispersal polymorphism that expresses itself as either long or short-winged individuals. These polymorphisms are easily observable, and more importantly, the short-winged phenotype is rendered completely flightless. This being said, dispersal and colonization is thus limited to only the long-winged insects, creating a dynamic in which spatial sorting can progress. Third, wing morphology influences the ecologically relevant phenotype of beak length. As we can see in this graphic, the darker bar, which represents the long-winged individuals, have in general 10% longer beaks than the short-winged conspecifics. This means that the best dispersing individuals are more likely to have longer beaks than the non-dispersing individuals, and thus any founding population should result in a surplus of big-beaked individuals. And lastly, in the greater Houston area, we had a major natural perturbation of soapberry bug populations. In 2017, Hurricane Harvey dumped 33 trillion gallons of water on the Gulf Coast. Some locations in Houston received 130 centimeters of rain in less than 5 days. This resulted in extreme localized flooding, which reached over 10 meters in depth in some locations and lasted for up to 3 months. So as you can see here in a series of images across time of one of my soapberry bug field sites, in which you can see even at 2 months, it's still inundated with a great deal of water. And such flooding is a real problem for soapberry bugs as they lay their eggs in the soil at the base of their host plant. A survival assay that we conducted determined that 50% of eggs perish when submerged for more than 2 days, and 100% perish before the end of the 3rd day. In addition, these floodwaters and heavy winds dislodged many of the seed pods and washed away the insect's food source downstream, resulting in high rates of localized extinction throughout the area. So to test our hypothesis, does spatial sorting accelerate rapid ecological adaptation when ecologically relevant traits are linked to dispersal? We continued to monitor 20 previously inhabited sites, 13 of which experienced localized extinction. This monitoring effort occurred every 2 weeks for 2 years. When insects were detected, they were captured, morphological traits were measured in the field, and then they were immediately returned as to not inflict any artificial selection on the site's population. We then compared the divergence of traits between surviving populations and recolonizing populations. Using these methods, we assessed our hypothesis by testing 2 predictions. First, only long-winged individuals will recolonize previously extinct sites. And this is intuitive as short-winged insects cannot fly. So here I'm going to show you the data from the first prediction. In doing so, I'm going to show you a line plot where sampling location is delineated by different colors and shapes. The y-axis will represent the ratio of long-winged individuals, and along the x-axis, we're going to have standardized sampling time steps, which with each step representing a 2-week period that moves away from the hurricane disturbance, which is represented as the gray bar with a red dashed line. To the left of the bar, we are moving backwards in time, and then to the right of the bar, we're moving forwards. So first, I'm going to show you the results from 4 rain tree sites that experienced localized extinction. As you can see, before the disturbance, long-winged ratio were diverse but stable, ranging from approximately 60 to 90% long-wingedness. But upon recolonization, all populations shifted to 100% long-winged individuals. And this pattern persisted for the first 12 weeks after recolonization. This is approximately the time it takes for a subsequent generation of offspring to reach adulthood. After this initial pulse of long-wingedness, we have this large fluctuation that over time stabilizes once again. Interestingly, the populations that had the highest ratio of long-winged individuals prior to the disturbance were not the populations with the highest ratio after the recolonization. This pattern of long-wingedness individuals recolonizing was similar for the other 2 host plant-associated populations, as shown here with balloon vine on the right and soapberry on the left. In comparison, we utilize control sites of balloon vines and rain tree. These populations survived the hurricane intact and did not require recolonization. Unfortunately, we don't have any controls for soapberry, as this is a natural experiment and all populations went extinct. And what you see is that wing lengths are not affected by the disturbance, as they remain stable both before and after the hurricane. Our second prediction is that beak lengths will be longer on the new founding population than the previously extinct population, as strong dispersers will have longer beaks than poorer dispersers that remain behind. So to test this prediction, I'm going to show you data from all 3 host associations in the form of a bar graph, with the y-axis representing the mean beak length of insects, and the x-axis divided between individuals from the pre-existing population and the initial colonizing individuals. That is to say, the long-winged individuals during those first 12 weeks after arrival. And to not bombard you with too much information, I'm going to present females during this presentation, but I can confirm that the patterns that I am showing you are the same in the male data subset. And what we see, starting with the rain tree on the left, is the initial colonizing individuals have significantly longer beaks than the individuals prior to the pre-extinction. This is interesting because it's not a comparison of the entire population. This is figuratively apples to apples. Dispersing long-winged females appear to have longer beaks than the morphologically comparable counterparts that stayed put prior to their extinction event. This pattern is repeated in the other 2 host plants, as you can see in the yellow center graph depicting the soapberry, and on the green graph on the right depicting the balloon vine. And so it's clear to say that after colonization, during that initial phase, their beak lengths are larger than the pre-existing population. This, however, is not the case in our control group of plant associations. There was no significant difference between insect beak length pre and post-storm in populations of rain trees associated insects that were spared from the flood and did not go extinct. We also failed to detect a difference between extant balloon vine associated populations shown here on the right. So great, we see evidence of spatial sorting in these recolonizing insects, and we see that the ecologically relevant trait is being dragged along as a consequence of this spatial sorting phenomenon. But can we say, what can we say about this potential mechanism leading to localized rapid adaptation or maladaptation? As demonstrated in past studies by Scott Carroll in natural populations, and then later confirmed by Meredith Senzer in laboratory experiments, a host plant seed pod size exerts divergent natural selection on insect beak length. Historically, as I mentioned earlier in my talk, Scott Carroll described a rapid decrease in the mean beak length of the insects in Florida after the introduction of the novel host plant rain tree. Meredith Senzer, who happened to be a co-author on a really great talk by Anastasia Burnett earlier today, who was talking about actually dispersal herself, showed that she later demonstrated how these observed differences have eroded as the populations have become homogenized due to gene swapping as a derived phenotype. It has now replaced the ancestral phenotype. However, this is not the case in Texas, which unfortunately has been a region of this insect's greater distribution that the literature has overlooked, although it is my intent to correct this oversight in the near future. Just to remind you, each of these 3 primary host plants have maintained divergent host-associated phenotypes in the face of gene flow. Balloon vine-associated insects having the longest beaks as they provide the greatest depth to which the insect must penetrate to access the seed, and rain tree-associated insects having the shortest as they have the narrowest seed pods. And so here we're looking at a subset of the data. These are the mean beak lengths of short-winged female insects. Because they are short-winged, they could not have colonized as they are incapable of flight. Thus, these insects are made of a later generation than the initial colonizers. As before, the y-axis represents the mean beak lengths, and the x-axis is divided between before and after extinction. So therefore, in this analysis, included only the populations that experienced localized extinction. Each plot is different, is a different host plant association. Interestingly, we can see that regardless of host plant, beak lengths of short-winged females are longer after recolonization post-extinction. This means that the initial shift in longer beak lengths that was observed during the initial colonization was maintained over subsequent generations. Here, we have detected evidence that spatial sorting has driven trait-specific adaptation across all 3 host plant associations. But is this really adaptive? As you may remember, the introduction of rain tree as a novel host resulted in a decrease in beak length due to natural selection as a result of its seed pods. Here, we see that the insect beak length instead increased and was maintained for multiple generations on rain tree in the face of strong opposing selection. Thus, spatial sorting has led to maladaptation of the soapberry bug on rain trees. On balloon vine, ancestral beak lengths were as long as 9 millimeters prior to the contemporary host shift, and subsequent gene swapping from rain tree associated populations. So here, the increase in beak length as a result of spatial sorting is instead adaptive as it begins to return the beak length to their ancestral norm. This is also true in soapberry associated insects. While early century museum collections are sparse for this association, short-winged female insect beak lengths were nearly a millimeter longer than contemporary lengths, making this increase adaptive to newly colonized host associations. So, in conclusion, we can say that 1, spatial sorting is occurring during recolonization, as we have shown that the frequency of wing phenotypes undergo an immediate shift due to differential dispersal, as shown here in our side-by-side comparison of rain tree associated populations that either went extinct and recolonized, or long-winged individuals versus extant sites where there was no shift in the wing length. Or wing polymorphism, Second, we showed that beak length, which is an ecologically relevant trait that is an independent of dispersal ability, is being dragged along as a consequence of spatial sorting. Again, we use this comparison of extinct and extant rain tree sites to show that there was a shift in beak phenotype as a result of recolonization by better dispersers with longer beaks. Third, we demonstrated that when spatial sorting does drag non-dispersal related traits, it has the ability to overcome opposing natural selection over multiple generations, although we do not yet know how transient this effect is at this time. And then lastly, spatial sorting has facilitated rapid ecological adaptation in balloon vine and soapberry, but has led to maladaptation in the rain tree associated insects, which adds like a really interesting dynamic to an already exciting and powerful study system. And so with that, I'd like to thank my collaborators, Scott Carroll, my advisor, Scott Egan, my lab mates. I would also like to express my appreciation for all the crew, and undergrads and high school students that aided in data collection. And a big thanks to my funding sources that supported this research, the NSF Rapid Grant, authored by Scott Egan and Scott Carroll, my independent grants, the American Philosophical Society's Lewis and Clark Explorer Grant, the Society of for the Study of Evolution's Outreach Education Grant, which allowed me to partner with research with the local high schools, and the American Museum of Natural History's Theodore Roosevelt Grant. I'd also like to take this moment to take a quick shameless plug that I'll be defending this upcoming summer. I'm currently looking for postdoctoral opportunities, so if anybody has any leads, I would love to chat with you afterwards. And with that, I'd like to thank you for listening, and I look forward to answering any of your questions if we have time.
All right, great talk, Matthew. We already have a first question from Andrew, who says, how can you separate spatial sorting, sensor shine, versus phenotype habitat matching?
Yeah, that's a really great question, and it's something that we've been working on, right? Trying to tease apart. So part of the problem is that one, we see that it's not actually matching, right? Because we have this maladaptive trait where they're not going to the correct tree with their beak lengths. They're actually bringing these longer beaks with them. And also we can't really, we don't see that when they're not being recolonized, right? So you're not seeing individuals coming into these patches where there's still populations, and then we don't see a shift where like natural selection is driving this to better match these host associations.
All right, maybe no more questions? Cool. Well, thank you very much, Matt, and thank you very much for all the speakers. I would like to remind everyone to join us for the Inspire Plenary by Dr. Scott Edwards in 1 hour's time. And thanks again for everyone that helped run this. Thanks, Volker, for doing the questions, and thanks, Mike, for doing all the technical stuff. This went really great. Thanks, everyone.